Okay, good morning, everyone. Thanks, Visha. Yes, my name is James Polli. I'm a faculty member at the University of Maryland. So welcome, for those of you who haven't been here before, welcome to the University of Maryland Shady Grove campus. I'm co-director with Anna Schwenneman for the CRCG, which is sponsoring this along with, of course, the FDA. And of course, we're here all in person here in Shady Grove, as well as usually a couple hundred people, sometimes even over 1,000 people online, to talk about updates on approaches to acceptable intakes of nitrosamine drug substance-related impurities, NDSRIs, and bioequivalence assessments for reformulated drug products. Okay, so the CRCG, the Center for Research on Complex Generics, I know some of you are highly familiar with it. For those who are not, just very briefly, it started a few years ago. It's a collaboration between the University of Maryland, Michigan, and the FDA. And our mission is to increase access to safe and effective generic drugs through enhanced infrastructure, communication, education, and research collaborations across industry, regulatory agencies, and academics. So those are the three buckets that we think about. This I know many of you are familiar with JDUFA funded research, and this was brought on board to complement that as a way to we do a lot of outreach is what we try to do. For example, we're wrapping up our second survey of the industry in terms of, industry as well as the public in general, as far as topics that are relevant in terms of complex generics and barriers and that sort of thing. We'll be publishing our second survey very shortly. And of course, we've had lots of workshops, and we also do have some research on the go, which there's a slide that lists those. So again, just much thanks to all the generic drug companies that are willing to interface with us. We're here to try to promote the development of complex generics. Okay, so this is a bit of a follow-up in a way to a CRCG meeting about a year and a half ago in the summer of 2023. And that was a real pleasure for me because that's where I first, I think, met many of you. And I would just encourage you, particularly for those of you online who are less familiar with CRCG, it's complexgenerics.org is our website. All of our training materials are there, including, for example, all of the recordings and slides from this prior workshop. Okay, since that time, a lot of things have gone on, including, this is just a slide showing the September CMC-related updated guidance, which will be the subject of course, a lot of discussion in the second half of this workshop. The first half, of course, will be talking about more safety issues, acceptable intake, and not shown here is a very important guidance that was put out shortly after our last workshop. I think it was probably about August of 2023 when that guidance was put out. So we've done lots of workshops, and we look forward to doing one in this very same place as well as online with regard to navigating the transition to low global warming potential propellants. And then we do have a schedule now for 2025. The first two are workshops, but there's an interesting aspect to them. There's also a hands-on demonstration regarding techniques with regard to in vitro permeability testing for topicals, particle size 1 in April, and then in the fall, in October and November, we have workshops on modeling and AI, as well as ophthalmic type products. Okay, so we do have some research on the go. I'll just mention maybe towards the bottom there, you can see that we've done some work on permeability assessment impacts, or actually no impacts of antioxidants as it turned out, as well as mitigation strategies. That will be certainly a topic of discussion, I think more tomorrow. And just on behalf of myself and the whole team, Anna Schwendeman, Visha, Dana, and Jennifer, and I'm really delighted that Dana is here. Normally you see her online, but I know many of you have interacted with her, and you'll be able to actually meet her in person if you're here today. And again, just would suggest to you to visit our website every once in a while. We do, if you want to join an email list, please do so. We send out probably about one email a month, hopefully not too much. Again, thank you to the planning committee. This is just a list of, I think, of the FDA folks Much thanks for Alan, who's done a lot of heavy lifting on putting this workshop together, as well as many of our industrial partners were significantly contributing to the scope of the workshop. Okay, and thank you for your participation. There's time for questions at the panel discussions. And for folks online, we also encourage you to ask questions. We do have a subject matter expert that will be looking at questions online. We want to be sure to include those. And maybe the most important thing I can say is all of this will be available in 3 weeks. If you registered for the meeting, you will get an email from us in about 3 weeks when things have been posted online. Thank you very much. And it is my pleasure to introduce Rob Lionberger. I think everyone probably knows Rob. Dr. Lionberger serves as the director of the Office of Research and Standards in OGD. He leads OGD's implementation of JDUFA science and research commitments, including internal research and external research grants and collaborations. His office provides pre-submission advice on complex generics through pre-ANDA meetings, product-specific guidances, and correspondence replies. Rob.
All right, thank you very much, Jim. It's fantastic to be here. I think the CRGC is a fantastic group that's funded by FDA through our GDUFA Regulatory Science program to facilitate, interactions that accelerate and make the development of generic products more efficient. And I think this is really a critical aspect of it. And so why are we here today talking about nitrosamines? And the reason is that through our regulatory science program, we ask industry for feedback in various mechanisms through the outreach that the CRCG does, through our annual public workshops, through our biannual meetings with the industry regulatory science work group that meets with FDA staff. We consistently hear that nitrosamines are one of the biggest scientific and regulatory challenges for the generic industry. And that's why, we had a workshop last year, we have a workshop this year. FDA is consistently coming out with guidances because we do hear this and understand how important this is to the generic industry. So it's certainly true that concerns about nitrosamines are not just limited to the generic industry. They also affect brand products as well. But we're here in this generic focus forum because the specific impacts on the generic industry are so extreme. And the way I think about it is that our system of pharmaceutical product development, regulation, brand products getting approved, making lots of profits, and then competition coming in to result in affordable medicines for the American public. This whole system, which is hugely successful, it generates innovation, it generates wide supply of affordable medicines at the end. It's a fantastic system. But the assumptions of this system don't really work when you get yourself into a situation like we found for nitrosamines, when late in the product life cycle, we're identifying new drug substance-related impurities, right? We really expect that if there are significant drug substance-related impurities, they're found the first time that product comes to FDA for review, right? That's the ideal. That's how the system should work. And when that doesn't work, then we get challenges. If these impurities are found early in the pharmaceutical life cycle, right, then appropriate pharm tox studies can be done as part of the development of the new drug programs. The costs for those then come from the profits that you make when you start selling those new molecular entities, right? And only one company has to do that. Generally, the generic products are more affordable because we don't ask companies to repeat pharm tox studies on impurities that are found in approved drug products, right? So if something's been qualified and there's a level that's available in a public product that a generic company can measure, we generally wouldn't ask them to do pharm tox studies for levels below that observed level. So that's sort of the foundation of our system. But when you bring in the new impurity late in the life cycle, lots of things go wrong, right? You say, well, now we're asking 20 companies to qualify it. Do they all repeat the same study? How do the generic companies pay for this? What's the cost recovery on this, right? And the usual way that they would look at it is say, well, it's in the brand product, But like I said at the beginning, this is also a product for the brand industry, so that's not a qualification level anymore as well. So the whole system really breaks down. And I think that's the challenge. But that's really why it's specifically painful to the generic industry. And some of the consequences of that can be, right, if it's too burdensome to mitigate these nitrosamines, right, generic applicants are going to exit from these products. That leads to shortage, that leads to less competition, that threatens the viability of some of the companies as well. So there's huge problems that come from that. So because of that, it's really critical to having a sort of sustainable, viable generic industry that we address nitrosamines in a sensible and practical way. And hopefully that's what we'll be doing here today. And one of the things that I absolutely love about the CRGC events is that they are very interactive. A CRGC event, to me, is not FDA getting up here and like telling you stuff and reading our guidance to you and telling you what to do. The CRCG events are really intended to have a wide variety of stakeholders and panelists. So we have FDA speakers, but we also have industry speakers, we also have academic experts. The perspective I hope that FDA staff take when they come to this event is we are here to learn, to listen, to use that to develop more effective and efficient regulatory programs going forward. That's, I think, the promise of the CRGC events. We have other FDA mechanisms where we do lecture you and provide information on our guidances and what we expect. But that's really not what I'm looking for here at the CRGC. I'm really looking for that interaction, that learning that helps us develop better systems. And I think today, really there's three aspects of this that I think we'll be talking about. We'll start out really talking about FDA's more recent guidance and updates on the acceptable limits, but we're also going to be talking about the more innovative approaches. We'll be talking about in silico methods to identify potential nitrosamine risk, talking about new assays and research underway on that, looking at the sort of the farm tox side, how do we do that more efficiently? Then we'll spend the middle two sessions of this meeting will be focused on mitigation, things we're doing. A lot of our experts at FDA lab have been doing experiments trying to say, what are the things you can do that can mitigate nitrosamines, right? Because that's really what you want to do. You want these not in your products. We've tolerated them for many years, but as we begin to recognize the risk, we want to find what are the best strategies to reduce them. And then finally, since FDA does actually really want to facilitate reformulations, new processes to get rid of nitrosamines, we have to make sure that our regulatory system allows you to do that in an efficient way. And we've made some definite progress in that in our most recent guidance that Jim mentioned. Right, for some BCS classes, we now say that for certain types of changes, you won't need in vivo BE studies. But certainly, we're also open to other discussions and other changes that go outside of that guidance scope. Because again, our purpose really is, reduce the number of nitrosamines, facilitate the reformulations that do that. And so you'll hear not just about FDA's guidance, but you'll also hear about space beyond the guidance. You'll hear about when you go into the science of the BCS class 4 drugs, right? What are the risks there? What are the risk factors there that are more subtle than the guidance? And this, I think, can help you develop, if you're trying to reformulate and you do have something that might be a class 4 drug, to think about the scientific approaches. And also, I think, one of the things that really struck me about our recent guidance is, it puts a lot of different regulatory requirements if you're sort of a BCS class 2 or class 3 or class 4 drug. And FDA, for BCS waivers, we've generally focused on, are you in class 1 or not? But really identifying very clearly those boundaries of those other products, not very clear. We recently, as you might have noticed, went through a global harmonization M13 for bioequivalence, and we updated about 800 product-specific guidances. So as part of that process, I asked my staff to tell me the BCS class of every one of those 800 products. And they went and they looked in the literature, and they went and they looked in FDA records. And we have a few guidances where we've said that they're class 1. But when you get to those other boundaries, there's lots of variation. There's this paper in the literature. It's, oh, this is a class 2 drug. No, it's a class 3. No, it's a class 4. So lots of thinking here. So again, that's the value of the CRG approach. Let's discuss the challenges and scientific aspects of implementing what we want to do with the common goal of better, higher quality pharmaceutical products. So with that, I think I'll be turning it over to Alam, who's on the organizing committee, to talk more specifically about this. But I will especially look forward to our final session. But I wish you all a productive and informative two-day session. So thanks to all of our organizing committee and the CRGC staff for doing this and making this event happen.
Okay, thanks, Rob. Oh, that's all right. So just to introduce Khondoker Alam, who's also known as Alam. He's a senior staff fellow in the Division of Quantitative Methods and Modeling in OGD's Office of Research and Standards. Alam is serving as a scientific lead for complex injectables, such as long-acting injectables and liposomal injections. He's also very much involved in mitigation activities, as we will discuss over the next 2 days. So I refer you more towards what's online as far as a broader CV, and I think all the other moderators would also appreciate being able to refer you to the CVs that are online. Alan?
Good morning, everyone. I'm Khondoker Alam. I go by my last name, Alam. I'm from Office of Research and Standards, and we are here today for our second nitrosamine workshop. And I'm very excited about this workshop. I have met many of you in virtually, but this is I'm meeting with you in person. I'm really excited to meet with you, and I hope that this workshop will be very successful. And I must say that we have come a long way with nitrosamine. Just a few years back, we had so many uncertainties about nitrosamine, how serious this issue is, what is the acceptable intake limit, what are the risk factors for forming nitrosamine, analytical method challenge, and how we will reformulate our drug product, how FDA will look into that reformulated drug products, bioequivalence study, should we conduct it or not. So these are all the uncertainties we had in our mind, and we felt that we need a platform where we can discuss this issue with our internal and external stakeholders. So with that context, we organized our first nitrosamine workshop last year, June 2023, and it was a very successful workshop. Our speakers talk about their latest research. We had a very good discussion, dialogue between our stakeholders, and that discussion actually helped us to develop our research priority and also updating our recommendation and planning for this workshop. So this workshop will download many FDA recommended guidance, and since the last workshop we have, I think that we have published two or three guidance, actually updated them. So we have lot of things to share this time. Our speakers will share that, and that's why we extended our workshop 2 days this year. And I must thank each member of the planning committee. This is a tremendous team effort, and it is a very truly collaborative effort between across different offices of FDA, and we have representatives from industry, academia, institutes, and CRCG members. They helped us a lot. Without their help, we cannot really organize this workshop. And I also like to thank Sam Rane, Fallon Small, Jennifer, Elise, and Chijang. Everybody really contributed to organize this workshop. So broadly, our workshop has 3 sessions. Session 1 is all about safety and toxicity related aspects. Session 2 is about product quality. What are the risk factors for forming nitrosamine, and confirmatory testing, analytical method, and risk mitigation, like adding antioxidant or pH modifier or other thing. And session 3 will be downloading our latest guidance, and we'll share our regulatory experience, how to address the pre and post approval changes. So to give you more update about the session 1, In this session, our speakers will talk about the safety testing methods of NDSRI and recommendation on acceptable intake for NDSRI. This is basically based on the carcinogenic potency characterization approach. You will hear about a lot about the CPCA and the tools behind this. We'll also talk about the future tools like quantum mechanics, how the quantum mechanics can really help us to complement the CPCA calculation. Our colleague from NCTR, they will be also sharing their updates on research on mutagenicity and toxicity. So you will get a lot of update from FDA, what is going on within FDA. But what about outside of FDA? So our speakers will talk about the research program that is going on under the umbrella of HESI, and you will also hear about Mutamine project that is funded by EMA, and we'll also talk about the nitrosamine risks coming from food. So you will hear a lot from all these aspects of toxicity and safety. It will be really a good session, I believe. Session 2, we have really versatile topics, and we will cover them in 2 sessions, subsession. Session 2A will be covered today afternoon, and session 2B will be covered tomorrow in the morning. Under session 2A, we'll talk about the risk factor for forming nitrosamine. The risk factor can come from API, excipients, manufacturing process, or even from environmental factor like water, air. So that will be really interesting talk. We'll talk about the mitigation strategy like addition of antioxidant, pH modifier, and the manufacturing process, and how we can use the kinetic model to assess the risks of forming nitrosamine. In session 2B, we'll be talking about current advancement of analytical method and what are the pitfalls or challenges of analytical method. We'll also talk about the API fragment NDSRI. This is a new topic. We all know that NDSRI is something that is very similar to API structure, but API can have that fragment, and nitrosamine can be also formed from the fragment. So this is our speaker will talk about the API fragment NDSRI and the extent of conversion and simulation-based risk assessment. And we'll also talk about the risk assessment methodology for NDSRI in the context of NAP test and modified NAP test. So this will be really good session. Session 3, I'll be moderating with my colleague Bingley. I'm really excited about this session. We have a lot to share. There are a lot of FDA speakers and industry speakers as well. And our speakers will dissect our current recommendation from the latest guidance. They will talk about the implementation on control strategy and the bioequivalence approach. We'll also talk about the implication of added antioxidant on drug permeability. Dr. Jim Polli will giving the talk. A little spoiler alert, we didn't see any impact. That is a good thing, I believe. I will appreciate that. And our speakers will also talk about the regulatory experience for pre and post-approval changes with relevant case studies, both covering with ANDA and NDA applications. So this will be very interesting talk, and I think that it will help drug developers to develop their own strategy. And we'll also talk about the risk management for BCS class 4 drugs products. So our latest guidance talked about a lot about BCS class 1, 2, 3, but how about the BCS class 4? So we'll talk about that, and we'll also talk about that how PBK model can be used for analyzing or assessing the risks or managing the risks. Few important things before I wrap up my presentation. So please hold your question till the panel discussion starts. And our panelists will address a question from in-person attendees, and then we'll go for the virtual question. And we may not be able to answer all the virtual questions because we may have probably more than 3,000 virtual attendees. So it's impossible, but please feel free to post your question, because we'll go through each question and your feedback and comments later, and that will really help us to understand that your concern or comment, what you have to share. And most important thing about this workshop, this is a purely scientific discussion. Whatever you hear from our speakers, panelists, and even from me, this is entirely from the scientific aspects, and doesn't mean to endorse any kind of idea. So without further ado, I like to welcome Dr. Bob Dorsam and Dr. Shinfu to roll up the session 1. Thank you.
Good morning. My name is Bob Dorsam. I'm Director for the Division of Pharmacology, Toxicology Review in FDA's Office of Generic Drugs. And I'll be moderating session 1 for today. We're going to be focusing on safety and risk assessment of nitrosamines in drugs. By way of introduction for the session, studies published decades ago tell us that nitrosamines can pose mutagenic and carcinogenic risk. But we know that the carcinogenic risk posed by these compounds is really compound specific. Although NDMA and NDEA had acceptable intakes established from robust published carcinogenicity information, similar robust data does not exist for many other nitrosamines that are detected in drugs. For some nitrosamines without carcinogenicity data, an acceptable intake has been determined by choosing a surrogate compound with robust carcinogenicity data based on some shared structural features. Surrogate molecules provide a viable method for approximating risk, but it's also apparent that the surrogate approach alone, based on a few dozen compounds with carcinogenicity data in published literature, is not sufficient to properly estimate the risk posed by many nitrosamines that are at risk of forming in drugs. This was especially true when we understood that nitrosamines might be quote-unquote small molecule like NDMA and NDEA, but they may also be structurally similar to the active pharmaceutical ingredient. These nitrosamine drug substance-related impurities, or NDSRIs, as we'll talk about them today, presented a new challenge in assessment of carcinogenic potency because there's no robust published carcinogenicity data for these compounds. This raises some key questions. First, how can we identify acceptable intakes for nitrosamines when we don't have robust carcinogenicity data? And then second, what are the appropriate in silico, in vitro, and in vivo studies that we can use to assess mutagenic risk of these compounds? Certainly, there's existing guidance, which you've heard about earlier, for NDA applicants, for ANDA applicants to use to address some application-specific issues, but some technical areas warrant further investigation. Many groups have invested significantly into the science that addresses these two questions, and today's session will provide updates from many of those groups where they'll provide a status update on their findings and ongoing efforts. Regarding the first question on how to identify acceptable intakes for nitrosamines when we don't have carcinogenicity data, one solution to this problem is the carcinogenic potency categorization approach, or CPCA, which is a framework that places nitrosamines into one of 5 categories of carcinogenic potency based on their structural features. The CPCA framework was a dramatic step forward, since it used knowledge of structural features for widespread use to estimate carcinogenic potency of compounds without additional empirical data. This approach is outlined in FDA's guidance, recommended acceptable intake limits for nitrosamine drug substance-related impurities, and that was finalized in August of 2023. And the basis of that system will be discussed by Dr. Naomi Kruhlak in the first presentation. Dr. Jakub Kostal will then discuss how quantum mechanical methods might supplement or possibly extend the predictions in CPCA. Our second question is, how can in silico, in vitro, and in vivo assessments be used to assess mutagenic risk of nitrosamines? Our following speakers, Dr. Cross, Dr. Heflich, Dr. Bercu, and Dr. Schuler, will discuss their data investigating various in vitro and in vivo assays to characterize mutagenic risk posed by nitrosamines. Worth noting, some of the work today is a result of extensive collaboration across industry, academia, and regulators. The HESI collaboration is an effort you're going to hear about today from both Dr. Jakub Kostal and Dr. Joel Bercu. Within HESI, there are multiple work streams with subject matter experts and working groups for Ames assay optimization, exploring in vitro approaches, QSAR and quantum mechanical modeling methods, and in vivo follow-up. You'll also hear about the Mutamind project today from Dr. Kevin Cross. As you heard, Mutamind is funded by the European Medicines Agency with multiple efforts, including on endogenous formation of nitrosamines in conditions similar to the gastrointestinal tract, on metabolic activation of nitrosamines, on DNA adduct formation, and on optimization of the Ames assay. Finally, in these workshops and in various sessions, the question is raised about how exposure to nitrosamine in drugs compares to nitrosamine exposure in foods. Dr. Anna Christodoulidou will address work from the European Food Safety Authority on the issue of nitrosamines in foods. We have an action-packed session, plenty to talk about, and so I'll introduce our first speaker. First up, we have Dr. Naomi Kruhlak, who is scientific lead for the Computational Toxicology Consultation Service in FDA's Division of Applied Regulatory Science. As a general, okay, Dr. Naomi Kruhlak.
Thank you, Bob, for the introduction. It's my pleasure to be here today, starting off today's session on nitrosamines and giving an overview of the carcinogenic potency categorization approach. I'm actually going to begin with a little bit of a recap of FDA nitrosamine guidances, just quickly touching on our publication timeline and different methods to determine acceptable intake limits, before diving into the CPCA, which was the result of a regulatory research, collaborative research project between FDA and other international regulatory authorities. So I'm going to go through some of the details of the CPCA, including its mechanistic foundation, the training set that was used to develop the model, how it was refined, how it was implemented, some predictive performance characteristics, but also some structural limitations. And then I'll finish up by touching on some calculators that are now available to calculate an acceptable intake limit using the CPCA based on chemical structure. And then I'll finish up by touching on some ongoing research in the area of structure-activity relationships and some future opportunities. So let's start with FDA guidances, and I'm sure everyone's very familiar with this, but I thought for completeness it was appropriate to acknowledge how long this issue has been around, which actually is really not that long, with nitrosamine impurities first being identified in June of 2018 with NDMA that was discovered in valsartan, but that of course quickly expanded to other drug products that had NDMA, and then also to other nitrosamine structures that were identified as impurities. This led to international collaboration among drug regulatory authorities to develop and harmonize approaches for addressing nitrosamine impurities, both from a quality and a safety standpoint. And of course, nitrosamine impurities are members of the cohort of concern as defined by ICHM-7. And as a result, they're regulated more tightly than typical mutagenic impurities because of their potential for high carcinogenic potency. And it has been estimated that between 25 and 41% of active pharmaceutical ingredients have some risk for nitrosamine formation due to the presence of secondary or tertiary amine centers in those API chemical structures. So in response, FDA published its control of nitrosamine impurities in human drugs guidance in September of 2020, which was then subsequently revised in February of 2021, to include steps to detect and prevent unacceptable levels of nitrosamine, as well as to identify conditions where nitrosamine formation could occur. The initial guidance included acceptable intake limits for 6 small molecule nitrosamines, and in the appendix for the guidance, there was information on how to calculate an AI limit from compound-specific data, but also by read-across from a structurally related surrogate molecule. And then this was followed up in August of 2023 with FDA's recommended acceptable intake limits for nitrosamine drug substance-related impurities guidance, also known as the RAIL guidance. And this was where the carcinogenic potency categorization approach was introduced in the US. It recommends read-across or experimental testing as alternate approaches after applying the CPCA. And it included the publication of a guidance webpage for a number of pre-calculated hypothetical NDSRIs where acceptable intake limits were presented based on the CPCA. And then most recently, in September of 2024, the control of nitrosamine impurities in human drugs guidance was revised again to expand the scope to cover not only small molecule nitrosamine impurities, but also NDSRIs. It also provides updated information on root causes of nitrosamine formation, detection, and mitigation. And it also introduces a flexible approach for calculating AI limits when more than one nitrosamine impurity are present. It presents some revised dates for risk assessment and reporting, manufacturing changes to the agency. And also notably, it includes a revised nitrosamine guidance webpage, which now covers acceptable intake limits, not only for NDSRIs, but also small molecule nitrosamines all in one place. So just to kind of show from a structural standpoint the difference between these small molecule nitrosamine impurities and NDSRIs. When we think of small molecule nitrosamines, we're talking about low molecular weight molecules. Often they're very simple molecules, maybe only 1 or 2 functional groups. In many cases, just simple dialkyl nitrosamines. In contrast to NDSRIs, which are often nitrosated forms of an API itself or a fragment of an API, and they have much more complex structures, many more functional groups, and are generally of much higher molecular weight. From a risk assessment standpoint, we need to be able to determine mutagenic potential as well as carcinogenic potency for basically any of these structures. So this is a fairly broad area of chemical space that we would need to be able to make these types of assessments in. So how do we go about determining acceptable intake limits? Per ICHM7, if we have compound-specific data, we have data for the compound, the nitrosamine impurity that's been identified, we can calculate a human acceptable intake limit by linear extrapolation from a rodent TD50 value, which is the dose rate at which 50% of animals in a long-term repeat dose carcinogenicity study exhibit tumors. So we can convert that 1 in 2 tumor incident rate in a rat to a 1 in 100 excess cancer risk in a human. There are rodent TD50 values available for about 65 nitrosamines in the carcinogenic potency database, which was developed by Lois Gold and capped several decades ago. This database is now available through the LASA carcinogenicity database with a structurally searchable interface. However, the challenge here is that many of these studies are not robust, and furthermore, most of these studies are for molecules that are very small, simple nitrosamines and not NDSRIs. Typically, when it comes to NDSRIs, we have very limited mutagenicity and carcinogenicity data available for them. So this is what leads to using an alternate approach, and the initial approach that was used, as Bob had mentioned in his introduction, was a read-across, where we can use robust carcinogenicity data for a structurally related surrogate molecule and read it across, apply it directly to our query molecule for which we have no or very limited data available. When we do this type of an assessment, we typically would select a surrogate based on the structural environment of that surrogate around the N-nitroso group, because this is the part of the molecule that's really going to have the greatest impact on carcinogenic potency. So these are some of the factors that we might consider when selecting an appropriate surrogate for our query molecule. And this is an example of a surrogate, NNK, that has been used to read across to several NDSRI structures. So that AI limit from the surrogate is then applied to the NDSRIs. However, there are several limitations to read-across analysis. Certainly one of the most notable limitations is that there are only a limited number of adequately tested surrogate molecules. And in particular, in some areas of chemical space, there really are no examples of surrogates that are available that are highly robustly tested. Also, these surrogates that we do have data for tend to be small molecule nitrosamines, so they're not necessarily going to reflect the complexity of an NDSRI chemical structure. Another challenge is that the selection of the surrogate can be a very subjective process, and it typically requires a fairly in-depth knowledge of nitrosamine structure-activity relationship patterns, including metabolism. And a common question that comes up is how similar is similar enough when selecting an appropriate surrogate? So this leads to it being a very labor-intensive process. So there was really a need to have some sort of prediction method that allowed us to leverage chemical structure, but where we were not going to be reliant upon data for a single compound. We wanted something that would better reflect the structural and mechanistic complexity in an NDSRI structure, something that's less subjective and more transparent, and something that just generally takes less time to implement. So this is what led to the development of the carcinogenic potency categorization approach. So the CPCA was developed through an international collaboration between scientists from drug regulatory authorities that are members of the Nitrosamines International Technical Working Group, or the NITWG. The CPCA was published in the respective regulatory guidances from those member organizations in July and August of 2023 as part of a framework for assessing acceptable intake limits. The manuscript to describe the scientific basis of the model, including its supporting data, was published in June of this year. The CPCA is a structure-activity relationship model for recommending acceptable intake limits that can classify an N-nitrosamine structure into one of 5 predicted potency categories that reflect carcinogenic risk. The categories have AI limits that range from the class-specific limit in that respective regulatory region, so in the US that would be 26.5 nanograms per day, up to the threshold of toxicological concern, which is 1,500 nanograms per day per ICHM7. The CPCA has its foundation in the alpha hydroxylation metabolic activation pathway, which is shown here. And this is the pathway that's responsible for the highly potent mutagenic and carcinogenic response that's seen for many nitrosamines. And in short, the step that is pivotal is the abstraction of the hydrogen in the alpha position, alpha to the N-nitroso group, and then subsequent hydroxylation, which then leads to formation of a diazonium species and/or a carbocation species that can subsequently form an adduct with DNA and lead to a mutation and subsequently potential carcinogenesis. So structural features that we know directly impact this pathway are included in the CPCA as part of that structure-activity relationship model. And therefore, we can generate a prediction of carcinogenic potency by considering the combined activating and deactivating effect on that N-nitroso group of those structural features in the molecule. The advantages of using this type of approach is that we don't need to have that compound-specific data in order to establish an AI, but we also don't need to identify a structurally related surrogate molecule which has robust carcinogenicity data, but rather we can leverage a broader data set of carcinogenic potency data. The CPCA was developed from a training set of 81 nitrosamines with mostly rat carcinogenicity data, supplemented with mechanistic information on metabolism and chemical reactivity. There were 3 types of data that were included in that training set. There were TD50 values that came from the carcinogenic potency database. There were some relative carcinogenic potency categorizations that had been published back in 1979 by Rao et al. And then also there were some negative classifications from the CPDB and also other publications in the area. The model itself was developed by visual inspection, so this is by manual interpretation of the data and extraction of patterns. And we incorporated some existing structural patterns that had already been published in the literature, but also incorporated some new ones that we identified through the course of our research. Each of those patterns were then assigned a weight to reflect the importance that they have on modulating that carcinogenic potency activity. So ultimately, we came out with 3 types of structural features or descriptors that were included in the CPCA. The first is the alpha hydrogen count and distribution, which reflects the immediate number of hydrogen atoms available on those 2 alpha carbons in the molecule, and we used a nomenclature where we show the lowest number and then followed by the highest number. And then there were 2 other descriptor types, a set of deactivating features and also those that are activating, those that increase potency. The alpha hydrogen score was assigned to the various different combinations of alpha hydrogen counts across the different atoms. And the score really reflects the potential for metabolic activation at that position. So cases where we have a low degree of steric hindrance and we have a higher expectation for metabolic activation will get a lower hydrogen score, which denotes higher potency. And then as we go to higher degrees of steric hindrance, we get a higher score, which indicates potential for lower potency. There were a number of deactivating and activating features. I should point out mostly deactivating features that were identified through the course of this work. And the resulting scores that were assigned to those features are shown here, ranging from +3 on the scale, on the end of strong deactivators, all the way down to -1 for activating features. The model itself is implemented as a flowchart, and this is really to make it accessible to anybody. There's not really a need to have a software tool to actually do the calculation. This can be done manually, so it's very transparent. It should be very easy to apply. The first part of the flowchart is actually dedicated to specific features that have such a strong deactivating effect that, if present in the molecule, will automatically result in it being assigned to the lowest potency category. So the highest numerical category, category 5, but with the highest AI, denoting a lower potency. So these include cases where we don't have an alpha hydrogen that's available for abstraction, so we wouldn't expect to proceed through that alpha hydroxylation mechanism. Also cases where we have a high degree of steric hindrance, again, which makes that abstraction of the hydrogen and hydroxylation step very unfavorable. And then lastly, cases where we have a tertiary alpha carbon, which even though we may in that molecule have the ability for the alpha hydrogen on the opposite side to be metabolically activated, it leads to a sterically hindered diazonium species that's likely to be detoxified through a hydrolysis step. But for any other molecules that don't contain those highly deactivating features, this is when we then look more broadly and calculate a potency score for the molecule based on all of its structural features. This potency score then is defined as the sum of the alpha hydrogen score, the deactivating feature score, and the activating feature score. So just to walk through an example calculation with N-nitroso-gatafloxacin. This is a case where we have a 1,2 alpha hydrogen environment, so we easily skip through those first 3 steps that we're looking for those very strong deactivating features. So we now get to the step where we need to calculate the overall potency score. We get a score of 3 from the alpha hydrogen environment. We get a score of +2 from the N-nitroso group in a 6-membered ring. We get a +3 from the carboxylic acid group, and we have no activating features in the molecule. So our potency score ends up coming out to being 8, which according to our flow chart would place the impurity into potency category 4 with an acceptable intake limit of 1,500 nanograms per day. There were a couple of different ways that we looked at the predicted performance of the CPCA. The first was to examine how well the model fit the compounds in the training set for which we actually had TD50 values. There were 58 examples that we worked with, and this plot shows those TD50 values against the potency categories. And we can see that we do effectively partition those compounds based on predicted carcinogenic potency. We see a stepwise increase in the median value within each of those ranges for the potency categories. But another way to look at the data is to consider compounds in the training set for which we had a categorical carcinogenic potency classification. So this is, again, from the Rao paper or the negatives that came from Cross and Ponting and other publications. So in this case, if we look to the left-hand side of the table, we can see that compounds that are known to be strong carcinogens or carcinogens are effectively placed in the most potent CPCA category. So the model is identifying them appropriately. It's very sensitive to identifying these strong carcinogenic species. However, on the weaker side of the scale, so these are the columns now on the right-hand side, we see that for compounds that have been experimentally determined to be weaker or even non-carcinogens, in some cases, the model is over-predicting. So it's leading to a more sensitive and possibly false positive result than it should. And this is somewhat by design. The model is not perfect, but the expectation is that if it's going to face a data gap where we have some sort of limitation in our knowledge, we would rather over-predict, under-predict, with the expectation that follow-up testing or other types of methods could then be used to justify a higher limit by demonstrating lower potency. So from a safety standpoint, that's kind of an appropriate way to be handling our initial screen, which is essentially what we're doing by applying the CPCA. There are some structural limitations to the model. It's designed to be applied to mono and dinitrosamines that bear a carbon atom directly bonded to the N-nitroso group. But it's not designed to be applied to other types of N-nitroso compounds, such as the ones shown here, N-nitroso-guanidines, ureas, and carbamates, where that alpha carbon is double bonded to a heteroatom. Another subclass of N-nitroso compounds where the CPCA does not apply is cases where that N-nitroso group is bonded to a nitrogen within an aromatic ring. The reason for these exclusions is because the expectation is that other mechanistic pathways can occur in those other classes. And therefore, using the alpha hydroxylation step, which is the foundation for the CPCA, would not be appropriate when applying it to these other classes, where in fact, some of them may be metabolically activated, or actually may be activated without needing that metabolic activation step. They may be direct-acting mutagenic species. So the CPCA was designed to be implemented very simplistically. It can be done as a back of an envelope type calculation by looking at the chemical structure and identifying features. But there are methods now that are available that automate this process. There are CPCA calculators that can take a digital structure, like a ChemDraw file or something similar, and then calculate the acceptable intake limit based on the CPCA from that structure directly. The benefit to having an automated calculator is it does ensure consistency across users that may have varying degrees of comfort level with working with chemical structures, but it also enables rapid processing of large numbers of multiple structures if there was an interest in screening them for potential risk. There are several commercial tools that are now available to do this calculation. We also, I should just point out, published a JavaScript along with our CPCA manuscript that does allow that calculation to be done automatically. It's not a standalone software, but it at least has the code that defines the features that we used. But there are also some lookup tables that have been published both by FDA and other regulatory authorities. In the case of FDA, we published acceptable intake limits for observed but also hypothetical nitrosamine impurities, and they're available on our guidance webpage. So I'd like to just finish up by acknowledging some ongoing research efforts beyond the CPCA. A lot of work has been undertaken through the Health and Environmental Sciences Institute, HESI, through their nitrosamines research program, which started with the three first work streams that are shown here, focused on Ames testing and then other in vitro testing strategy and in vivo follow-up. But more recently, a new work stream was introduced with a focus on QSAR and also quantum mechanics. And some of this work will be covered by our next speaker, Jakub Kostal. The intent of this work is really to further refine and extend the CPCA using these additional methods, so using quantum mechanical calculations, with a focus on certain types of structural features that are in the CPCA, including those that are electron conjugating and withdrawing, those that have ion stability considerations, and also ring features. So I think in general, there are some future research opportunities for expansion of the existing CPCA framework by leveraging some of these newer methodologies, quantum mechanics calculation, but also to take advantage of some of the work that's going on in the area of metabolism modeling and prediction. And I think this can be facilitated by using some of the newly generated experimental mutagenicity and also metabolism data that's being generated by different groups. There's also an interest in research into when and how a read-across analysis can be used as an alternative to a CPCA. So looking more into which classes of structures where this read-across approach may be most appropriate. And potentially identifying and sort of tightening up, quantifying some of the key parameters that could be used to support selection of a surrogate to make the process perhaps a little less subjective than it currently is. And then lastly, I think there's some interest in exploring structure-activity relationship modeling for other N-nitroso compound classes that are currently out of scope of the CPCA, and these would be the N-nitrosamide-like compounds that I mentioned on one of my earlier slides. And with that, I would like to acknowledge members of the Nitrosamines International Technical Working Group for developing the CPCA. Also my FDA colleagues in computational toxicology and pharm tox, and thank you for your attention.
Thank you, Dr. Kruhlak. So next up, we'll have Dr. Jakub Kostal, who will talk about the QSAR-QM HESI project. Dr. Kostal is Associate Professor of Chemistry and Director of Environmental and Green Chemistry at George Washington University and co-founder of ToxFix. Welcome, Dr. Kostal.
Good morning. All right, so as Naomi mentioned, I'll be pivoting towards looking at quantum mechanics, how we can use these sort of newish approaches to both enhance the CPCA, perhaps validate some of the features of the CPCA a little better, and then talk about how QM on its own can advance in silico or advance hazard assessment of nitrosamines. So just starting very broadly, so when you look at in silico tools, the landscape of in silico tools in predictive tox, you have, and this is contentious, but broadly speaking, you have these three basic categories. You have your QSARs, you have your expert systems, and you have your read-across. Now, historically, we have been assessing compounds for hazard using these three categories at the atomistic level of molecules, right? So we're looking at the atoms in the molecules, their connectivities, and from that we can derive similarities that then help us figure out hazard potency or just hazard category. So that's the traditional interpretation. Now, what's neat about quantum mechanics is that you go below that atomistic level, you go to the electrons, right? And if you think about chemistry, fundamentally, chemistry is just a flux of electron density in particular environment. So you can make the argument that while the atomistic approach is fast and useful, the electronic approach can give you a higher resolution and a more fundamental tie to the chemistry that's underlying the relevant metabolic processes, right? And it can be used in all those three categories. So you can use quantum mechanics in a QSAR system, you can use it in an expert system, you can use it to do sort of electronic structure read across. And because it's fundamental ties to the underlying chemistry, you can make the argument that it minimizes the Achilles heel of all the atomistic approaches, which is training set bias, right? So training a model that's biased on the compounds for which you have data. A perfect case, nitrosamines, where you have available data for very small compounds that are quite different from the compounds that you want to test on. So there's an argument to be made that quantum mechanics perhaps can give you a greater insight into the behavior of those larger compounds. All right, so bear with me. I'm going to try to summarize all of quantum mechanics on one slide in 1 minute to a diverse audience. So I'm going to inevitably fail. But the way I like to think about it, and maybe I should start with a statement that quantum mechanics is not a monolith, right? It's a field of approaches with different trade-offs, approximations, picking different levels of theory to address different problems. So it's a very diverse field. Now, in terms of the diversity, the way I like to start is to think about the system size. So for nitrosamines, you can start with the single molecule of the nitrosamine, which you see on the left there. You can also increase the system size and consider the effect of the environment, right? So you can throw your nitrosamine in the water and model the properties of the nitrosamine with regards to that environment. And you can extend it even further and consider the biological targets. So in this case, you can consider the structures of CYPs and how those structures, how that heme group might affect, right, the transformation of the nitrosamine deactivation to the diazonium, right? So you can kind of grow your system size. Now, in terms of what you can calculate, again, this is a horrible oversimplification, but on the simplest level, you can calculate global properties, sort of the electronic properties of the whole molecule with quantum mechanics. But you can also go a little more refined and look at specific locations in a molecule, specific reactive centers, and look at their electronics and look at their sterics. And then one step below that, you can actually carry out explicit molecular interaction simulations. We so-call these reaction pathway modeling, where you would take a known chemical process that may underline the transformation that then leads to the toxic outcome, and you would essentially mimic that process that you drew in an atomistic sketch using quantum mechanics, and you would calculate the barriers which relate to the kinetics of that process. You would calculate the free energy changes which give you the thermodynamics, and from that you would get sort of a very robust understanding of the propensity for that process to take place for any given molecule. Now, the more information you have about the metabolic process, the more you can go down this pyramid, the more you can look at very specific high-resolution events, right? Now, if you don't have those events, if you don't have that knowledge or description, then you're kind of stuck at the top, right? So you have to consider sort of the interactions or the behavior more globally, more non-specifically. The problem that we arrive at is that we always think of in silico tools as being cheap and fast. But when it comes to quantum mechanics, cost does become an issue. So when you think of modeling single molecules like in DEA and just looking at properties, electronic properties for that molecule alone, so very globally, that can be a calculation that's done in seconds. But when you start getting more specific and you grow your system, you're talking potentially about months of calculations, which is of course impractical for hazard assessment. So there'll be some trade-offs that you have to take care of in order to make quantum mechanics useful. All right, so how does that all translate or apply to the nitrosamine landscape? So Naomi talked about the CPCA, which stands at sort of the top of the pyramid. So it's an atomistic approach, it's an expert system that gives you mechanistically justifiable rules based on features in the molecule to help derive the AI limits. Now, when you go into a more, or I would say higher resolution modeling, on one hand, you have the quantum mechanics approaches, which again, we can roughly split here into the reaction pathway modeling, which was the last triangle at the bottom of the pyramid, and quantum mechanics that deal with either global or local properties of the molecules. And those are typically integrated into some sort of QSAR, so a statistical model, which would then predict the outcomes. So I mentioned a few publications which I'll discuss in more detail just to give you a sense of the current landscape. The main utility of the reaction pathway modeling is that it's very useful to compare specific events across several different NDSRIs at very high resolution. So you effectively can do what I mentioned on my first slide. You can do this electronic structure read across, right, to potentially justify a specific limit for a compound relative to another compound. Now, as for the QSAR-QM, so using global and local reactivity indices that are fed into a statistical model, you can use these kinds of models to predict carcinogenic potency potentially in a robust way, as long as your QSAR model is properly validated and externally tested. Now, I want to briefly mention a complementary approach to the quantum mechanics here that, again, goes to enhance the current approach with the CPCA, and that's dealing with something that's omitted in everything that's been so far published and discussed on quantum mechanics for nitrosamines, and that's the effect of the active site. And for NDSRIs, the big question being, can the molecule actually fit into the SIP? And can it fit in a pose that's suitable for catalysis, right? So does it have the right orientation to start with the hydroxylation of the alpha hydrogen or the hydrogen of the alpha carbon, right? So we have these docking approaches that can potentially deal with this. And we have developed color coding schemes so you can get a sense of how likely it is for the compound to undergo a catalysis, fit into the active site, and fit in with an orientation that can potentially lead to the hydroxylation. All right, so now pivoting all this, just to give you a sense, so that was the landscape of QM for nitrosamine. So how does that relate to the sort of HESI QM QSAR goals, which again, main objectives of those goals is to help validate and strengthen the CPCA rules and potentially deliver an expanded or augmented CPCA. Naomi mentioned the three sort of major goals, so looking at cyclic nitrosamines, looking at electron withdrawing and donating effects, looking at diazonium reactivity, and then more in the midterm goals, looking at the integration of the QM and CPCA directly. Now, the strategy for these, and I'm going to sort of give you a sense of it very briefly in the next slides, is to essentially develop new structure-activity relationship using the QM information, right? So the QM information, again, goes below the atomistic structure at the movement of electrons and using values of energies and whatever other reactivity indices that you get from the QM modeling to potentially derive new SARs. Now, just keep in mind that there are sort of two goals that can be accomplished in this process. One is you can enhance the CPCA, and you can also figure out how you can use QM as a standalone tool aside of the CPCA to enhance the hazard assessment. So just a few examples to give you a sense of what the group has been working on. This is a series of unsubstituted cyclic nitrosamines, again, growing in size. You can generally see that the TD50 decreases as you grow the size of the ring. Doesn't seem to be related to sterics, which would be the first instinct here. So we looked in our quantum mechanics calculations at various steps of the nitrosamine activation pathway. And the one step that seemed to be important is the heterolysis step. So this is a step where you have a proton transfer from the hydroxylated alpha carbon to the nitrosamine. Now, what we know about proton transfers, and this is getting into really cool but really wonky quantum mechanics. What we know about proton transfers is that they will have a barrier dependent on the distance between the acceptor and donor. So as the acceptor and donor get closer, the barrier gets smaller, so the proton transfer gets faster, right? Now, because proton is very small, it can tunnel through this barrier if the barrier is small and narrow, right? So it's something that you need quantum mechanics for to capture accurately. What we saw initially is, and this is, again, remember that system size arrow, we looked at nitrosamines in aqueous solution in water, is that there was a change in the distance between the acceptor donor as you varied the size of that ring. So we kind of carried out this calculation across the entire series for which we had TD50 data and saw that there's actually a really good correlation between the TD50 and the distance between the acceptor and the donor, right? So essentially correlating the barrier of that proton transfer, how kinetically favorable it is to the TD50. And it even captures, this is the neat thing, it captures kind of the fluctuations in the TD50 that you get in this series, right? Because the distance between the hydroxyl group and the nitrosyl doesn't necessarily go bigger or smaller with the size of the ring. The ring of different sizes will adopt different conformations that may change that distance. So it's important to kind of look at the 3D structures, look at their conformation landscape, and then use QM to better understand some of these processes. This is another nice example. This is from a paper by Lilly, who have looked at, it's a little fuzzy, who have looked at a series of nitrosamines that had either electron-donating groups or had electron-withdrawing groups. And they saw that the electron-withdrawing groups are less toxic. This can be justified using physical organic chemistry. We know that electron-withdrawing groups would destabilize the radical that's formed in the hydroxylation step, which would make that process less likely. Now, if you have electron-donating groups, then through sigma effects and through cool things like hyperconjugation or orbital mixing, you can actually stabilize that radical intermediate, and you make that metabolite more likely to form, so the parent compound can be deemed more toxic, right? Now, we can see this through mapping the transformations in the reaction pathway. So we actually look at the energy changes as we take the nitrosamine all the way to the diazonium, and we look at the reactivity with the DNA. Right? So the barriers in this process, particularly for the first and the last steps, confirm the hypothesis that electron-withdrawing groups are going to destabilize the molecule, make it less toxic, and so the barriers were noted to be much larger for this process than for the electron-donating groups. So these are the sort of SARs, right, that you get from quantum mechanics that then you can leverage to potentially get a higher resolution in the CPCA or validate existing features in the CPCA. Another example here from, again, work by Lilly is looking at different heterocycles of the nitrosamines, again, having some TD50 and AI values derived from those TD50 values. And here what seemed to be important is in distinguishing the carcinogenicity of these compounds is that last step, is that reactivity with the diazonium. And there was a nice linear fit across these compounds based on the barrier, again, the kinetics of that step. So again, an information that for a series of nitrosamines can give you a higher resolution into their toxicity. This is a paper by Pfizer looking at the diazonium reactivity in a different way, looking at actually the pKa, so the acidity of the diazoic acid. The stronger the acid, the more the equilibrium is pushed away from the reactivity towards the DNA, and that could be captured by simply building a univariate correlation between the pKa and the TD50 values. So large barriers, lower reactivity for heterocycles in the DNA conjugation step leads to lower toxicity, and the stronger the diazoic acid, that also leads to lower toxicity. Now, another way to look at this is to look at the length of the CN bond in the diazonium, where they noted that the shorter bond would lead to a more potent nitrosamine, again, generating a relatively well-fitted relationship. Now, those were just a few examples of how you could use SARs generated from QM to help with the CPCA, to help augment the rules in the CPCA or help refine them. Now, you can also use QM, right, for direct predictions of carcinogenic potency to set AIs. And in some ways, you can look at this as even the lower hanging fruit. So what I want to show you, there are more tools that are out there, but I want to show you two that have been published recently that demonstrate a capability that's already available now for assessment of NDSRIs. So one, again, is the paper by Pfizer, where combining some of those features that I discussed before, namely the CN bond length of the diazonium, the OH bond length, so this is the distance between the oxygen on the iron in the CYP and the hydrogen on the alpha carbon in the transition state, and then something they call GSS, which is a ground state stabilization, a thermodynamic quantity that looks at the size of the rings or the number of rotatable bonds in aliphatic carbons. And they could use these variables to build a strong correlation to TD50 values, which had about 0.8 or above 0.8 R squared. The caveat was that it was only applicable to smaller nitrosamines and nitrosamines that didn't have features that would lead to excretion, for example, carboxylic acid and so on. So we always have to pay attention to the applicability domain of any model to see whether it's useful for your particular compound. The other model that, this is our work that's published, is one that looks at it slightly differently. It's a lower resolution model. It doesn't predict TD50s, but it predicts categories of potency. And again, uses mechanistic features from the metabolic pathway of the nitrosamine activation to build relationships to the known nitrosamines with calculated TD50 values, looking at, again, the hydroxylation step, the heterolysis, which I described before, and then the diazonium reactivity. Models, just as the linear regression model from Pfizer, this model performs well as well. It's about 80% accurate in external testing, and it's also capable of distinguishing carcinogens from non-carcinogens with above 90% accuracy. And if you pair it with the docking that I mentioned on the second slide, so looking at, well, can these compounds actually fit into the active site? You start getting a more holistic picture of what kind of an AI you may want to set for your particular NDSRI. This particular model, lower resolution than the TD50 model, but applicable to all NDSRIs. So greater applicability domain. And those are some of those trade-offs that I mentioned initially in the QM approaches, right? Different approximations, different trade-offs, which lead to different applicability. All right, so this is my take. This is not, these are not thoughts of the whole HESI group, but in my view, the way we're currently in the in silico landscape for nitrosamines, we have our CPCA as a very useful initial triage tool, which we are hoping to augment, enhance with the QM calculations. Now that's useful in setting the initial AI limit. Then we have these statistical QM and potentially QM enhanced with P450 binding models, which can help us refine these AI limits. And then at the highest level of resolution, we have the reaction pathway modeling, which can give us a comparison between two or few NDSRIs to figure out which events in the metabolic pathway might be driving the difference between these compounds and potentially use that information to argue for a different limit. So we have these tiers of assessment that can be combined together to give you a much better understanding of the behavior of the compound and then what an appropriate limit might be. Now, that's all for me. Happy to discuss it later, but I'm going to leave you by a quote from Niels Bohr. So Niels Bohr, I'm sure all the chemists in the audience know, this is the first model of the atom, a nucleus with electrons orbiting at different levels. So this is kind of the foreground for quantum theory because every level has different energy. But the model itself, right, assumed that the electrons are particles. So Niels Bohr started as someone believing that the electrons are discrete particles. Of course, now we know that in quantum mechanics, there's this particle wave duality, which by the way supports that whole proton tunneling I said, or I discussed for the proton transfer in the heterolysis step. So we know that the wave nature of electrons is important. And if you look at Wikipedia page for Niels Bohr is actually credited as one of the fathers of quantum mechanics, someone who created a very classical model, but then changed his views, knowing that there is a better theory to explain the physical world around. So I'm going to leave you with that sort of a summary of progress being necessary and seeing change as inevitable part of that necessary transformation of the field. All right, thank you.
Thank you, Dr. Kostal. Next up, we'll have Dr. Kevin Cross. He'll be talking about the projects that are being conducted under the EMA's Mutamine project. Dr. Kevin Cross is head of science at InSTEM, and we look forward to hearing about the work under EMA's Mutamind.
Thank you, Dr. Dorsam, and thank you for inviting me to participate in this workshop. Before I start, I just wanted to provide a little addendum about what Jakub was discussing in terms of the Hesse work on quantum mechanics. I just can't resist because I'm working on that with him, and it's very exciting. But when Jakub talks about the QM enhanced approach to the CPCA, the idea is that we're going to create more thorough, simplistic rules for enhancing the CPCA, not requiring the CPCA to run quantum mechanics at that level. Okay, so I just wanted to make that clear. The other point too is that quantum mechanics, when we drill down and looking at the very small reaction sites and reaction features, is a very localized approach, but it also needs to be tempered with the whole molecule approach, such as looking at the solubility of the molecule, other global phys-chem properties, and those will affect their activity in real experimental work as well. So now we're going to transition from looking at the theoretical to the experimental, and the next 4 talks are going to talk about experimental results in testing nitrosamines. And I'm going to talk about the EMA Mutamind projects, which was a project of contract of 3 projects that was funded by the EMA over a course of 1.5 to 2 years, depending on which of the 3 projects it is. And this was a collaboration contract with a number of different companies headed by Fraunhofer Idem out of Hanover. And the principals on that were Sylvia Escher and Christina Ziemann. So the focus of this project was really 3 separate projects to better understand the processes involved in mutagenicity. We picked 40 nitrosamines with a special focus to try to look at bulky and drug-related compounds. So there are 3 projects. One is the endogenous project, which looks at endogenous formation of nitrosamines from APIs under realistic endogenous conditions in the gastrointestinal tract. The QSAR project had 2 goals, which is looking at metabolic activation of derived NIs to identify involved CYP enzymes, and also DNA addict formation and their kinetics and repair. And the third is the in vitro project, where we looked at optimization of the in vitro and mutagenicity assays, specifically Ames and in vitro COMET. And a little bit later, Mike Schuler is going to be talking about in vivo COMET. So, Dr. Bercu is going to be talking about the Hesse work on the enhanced Ames project. So something to keep in mind is this work was all going on while the enhanced Ames protocol, also called the EAT, was being developed. Okay, so I'm going to briefly just touch on the highlights from the 3 projects today, given the time that we have. The aim of the QSAR project was to investigate the metabolic activation, DNA addict formation, and repair mechanisms to distinguish different classes and looking at their different potencies. So this involved different DNA alkylation assays of primary hepatocytes or HepG2 cells, also calf thymus alkylation as well. Analytical work was LCMS or LCMS-MS to identify the alkylated nucleosides. So here in this particular project, these are the different types of compounds which were tested, the nitrosamines, some of them being more drug-related than others. And just a brief summary of what we found when we looked at the DNA alkylation process was that we saw, as expected, carcinogenicity potency was increasing as the O6 guanine to N7 guanine addict ratio was increased. The O6 methyl guanine formation of DNA strand breaks we found to be a later event whereas spontaneous depurination of adducts formed at N7G was an early event. So this was a little confounding when we first did the work because we weren't seeing any DNA formation because we hadn't waited long enough to look at the formation of 6-methylguanine. We also found that lower potency nitrosamine showed fewer DNA strand breaks, and that we really needed to look at MGM deficient cells and longer incubation times to substantially improve the predictivity of the carcinogenic potential using the in vitro alkaline comet assay. The second project is the in vitro mutagenicity project, which is really looking at optimizing Ames test conditions for mutagenicity, as well as evaluating in vitro comet assay with different liver cell models, including HepG2, primary rat, and human hepatocytes. Just to gloss over the protocol, we took an iterative approach to testing a set of nitrosamines in different solvents with different S9 mixes to understand the predictivity. We then optimized that protocol and tested additional compounds using some different solvents and S9 mixes before we finally came up with a final protocol, which we then used to analyze the predictivity of 13 nitrosamines. For the in vitro comet assay, I just wanted to mention again that there were several different cell tissue models for testing the nitrosamines. Only 10 nitrosamines were tested here. This included primary rat hepatocytes, human hepatocytes, FG2, and also liver slices, human liver slices. Okay, so just as a summary, we see the different tests that were run in terms of Ames and Comet for the different nitrosamines, categorized on the left-hand side by their potency as predicted by the CPCA. And we're not, saying that the CPCA is always going to give you the right predictions, and we're not convinced that necessarily the experimental results are always the definitive gold standard either, particularly for the in vitro assays for Ames and Comet. As we know, we can get more accurate results with in vivo results. But we found a good correlation here. We found some cases where it appeared in category 2 where there were some compounds that were not predicted Ames positive that we thought should be based on the CPCA approach. And we went to look at trying to understand why that happened. And we also found some cases where we found some Ames positives in the cases where category 5 CPCA predictions were made, where we'd expect low or no potency of those carcinogens. So in this work that was done, we used non-induced hamsters. And so that was one theory of why we had some cases where we saw what we would consider false negatives with the Ames approach that we used in our optimization. It also could be a factor of the characterization or lack of characterization of the S9's particular enzymes, CYP enzymes involved in the particular compound interactions. Okay, another thing we did though that was quite interesting is we actually looked at the response, the Ames response of the nitrosamines in the most sensitive strain and conditions and that was TA100 with 30% hamster S9 and 7 microliters of DMSO. And we plotted the BMD 100 values and just looked at their relative potencies and then found that there were basically several different categories and we could then relate those to the different CPCA categories. We actually found a pretty good correlation here between the potency as determined by, or it could just not potency, but the Ames response and the actual categories as predicted by the CPCA. There were some things that we expected, and one thing we didn't expect, for instance, a relatively low response for the last compound, which is a 2-methoxy and 2-methoxyethyl and nitrosoethylene. And this was a case where when we looked at it closer, we realized that this was actually another compound, Andella, which was had a couple of extra methyl groups on it, and those methoxy groups looked to be fairly labile. And we conjectured that there was probably some metabolism where the labile oxygens broke off, and actually you ended up with this compound metabolizing to Andella. That was one conjecture here. But we found this to be quite interesting because we know that the Ames test itself is not designed to look at risk assessment and potency assessment, but merely hazard assessment. So the overall conclusions from the sensitivity analysis were that solvent effects were really quite limited, particularly when small volumes of solvent were used, and in larger compounds and NDSRIs, we found it also to be quite limited in terms of affecting the response of the Ames assays. We found higher sensitivity and response using hamster S9, 30% S9 over 10%. There'll be more talk about this in the upcoming presentations as well. And we found out that the Ames response aligned pretty well with the CPCA categories. Lastly, I want to talk about the project on endogenous formation of nitrosamines. We're going to have another talk on nitrosamines in foods that's coming up a little bit later, and so this is a little bit of a segue into that, the work that we've done here, the EMA contract on this. And this was to investigate the impact of physiological conditions on the emergence of nitrosamines from an API, and particularly looking at the direct introduction of a nitroso group to the API molecule in endogenous conditions, okay? So the endogenous formation of nitrosamines both in the stomach and in the gut, the human biome. So the results of this from the impact on the gut bacteria on the formation of nitrosamines, we found that few APIs actually inhibited the growth of individual tested bacteria, and that pH values had to be controlled quite strictly in order to avoid spontaneous nitrosamine formation, which occurred at lower pH. So we had designed a instrument for cultivation of bacteria under anaerobic conditions, simulating the actual atmosphere in the gut, and use that for this process. If we see down here on the lower left, the ratio between spontaneous and bacterial-mediated nitrosamine formation, we actually see that there really is really none. In other words, we did not see any nitrosation of APIs in the gut when we introduced nitrosating agents into our simulated gut of, I think we had 12 different bacteria that we'd selected for this. So what we did see was reduction of nitrate in the presence of bacteria. The nitrate concentration rapidly decreases in the presence of the 4 bacterial species, including E. coli. The arrow there pointing to the nitrogen decrease, or the nitrate decrease in the gut. Actually, it was also twofold. It also showed, not here, but we showed that there's actually both conversion of the nitrate groups to the nitrite, and the nitrite groups to ammonia and nitrogen as a detoxification effect. They also fed the bacteria, so you see on the graph where you see an increase in the data points. That's actually the growth of the bacteria from eating the nitrite. So stomach nitrogen nitroso formation is different, obviously, from the more acidic conditions in the stomach. And here we did see a dependence on the nitrosamine formation by its protonation. And from this, we could look at estimating the protonation of secondary amines in the stomach by looking at the pKa of the APIs. Okay, and so this allowed us to develop a method where we could distinguish compounds based on based on their chemical properties as to how potent we would think these nitrosamines would be in the stomach, and how their formation would be driven. And as part of that, in the contract, a PBK model was developed, where we estimated the Cmax in plasma and liver, the conversion of APIs to nitrosamines under gastric conditions, as well as the transport of those to the liver. And this is another result of the contracts that we had. So that's a quick overview of a lot of information, but it's introduction now into some of the additional talks we're going to have on the experimental results from different groups, such as HESI and Pfizer. And I thank you for your attention.
Thank you, Dr. Cross. So now we will have Dr. Robert Heflich, who is Director for the Division of Genetic and Molecular Toxicology at FDA's National Center for Toxicologic Research. Dr. Heflich will discuss mutagenicity and genotoxicity evaluations of NDSRIs coming from his laboratory. Thank you, Dr. Heflich.
Thank you, Dr. Dorsam. Those of you who know me know I speak very slowly at times, so I need every second allocated to me for this talk. Let's see. There's a green button. Oh, I see, the big green button. As it says on this slide, I'm at the National Center for Toxicological Research, or NCTR, which is part of FDA. And if you don't recognize it, it's a non-regulatory part of FDA that specifically is involved with generating research data that is either requested or we think FDA should have for fulfilling their regulatory mission. So we became involved in this issue of nitrosamine drug impurities 4 or 5 years ago, and it's since grown to rather large proportions at our institute. The people on the left of this slide are all lab people who work at NCTR on various projects related to nitrosamine drug impurities. I've list 3 projects here. We also are trying to get a carcinogenicity project going to do the neonatal mouse assay. Hopefully that'll start this year, and we get some data that we think will fill in some blanks as far as NDSRI carcinogenicity data. On the right are listed people from the regulatory side of FDA. The PI for this project is Asa Estrachi, who pronounces her name a little differently, and I can never quite understand how she does it. But anyway, Asa has been the lead on this. It's fair to say, as far as keeping this project on track. So, maybe I should go back. I'd like to spend a little time on our AIM studies. They've reached sort of a plateau in that we've published most of what we have, so that's all available. And spend some more time on in vitro mammalian cell studies, where there are questions in my mind is exactly what we should do and where we should be going, and hopefully we'll get some feedback from this audience that might help that process. Okay, on to the AIMS test. What we were asked to do was to generate a method for doing the AIMS test that was the best, most sensitive approach to AIMS testing that we could think of. And we were also asked to find out something about the mutagenicity of NDSRIs. So we combined those two in this study. We looked back in the literature for methods that had been described that potentiate the mutagenicity of nitrosamines, small molecule nitrosamines in the Ames test, and trying to avoid those things that didn't work. And we applied these to those bunch of small molecule nitrosamines just to confirm that these things that were described did have an effect on sensitivity of the assay, and then applied those to the NDSRIs we were looking at to see how they responded to these protocol variations for the Ames test. And what we looked at is described in this slide. And as I said, this is all described derived from the literature and my understanding of what was important and what wasn't. We looked at 5 tester strains. We looked at 5 different types of metabolic activation, including rat liver S9 and hamster liver S9 at 2 different concentrations. We did the pre-incubation assay almost exclusively, and we did pre-incubations for 30 and 60 minutes based on some literature observations with small molecule nitrosamines. And we tried to avoid solvent problems by using a small volume of solvent. This is a particular problem when doing the pre-incubation assay that those of you who are familiar with these testing methods would appreciate. We wound up testing 12 small molecule nitrosamines and 17 NDSRIs using a standard set of conditions to look at all of them. So here's the list of compounds. So on the left is the small molecule nitrosamines. I've highlighted in yellow the ones that are also part of the HESI study. Many of these have cancer data associated with them. On the right are the list of NDSRIs we looked at. Only one of them has a report in the literature, but not in the LASA database of carcinogenicity findings. So here are some highlights from our study. I'm not going to present any data or anything. 18 of the 28 testable substances were positive under one condition or another. And I say 28 testable because one of them was so toxic we couldn't really test it and get meaningful data. Combination of TA1535 and the WP2 strain that we use with the PKM101 plasmid was able to detect all mutagenic nitrosamines, and a couple of them were specifically mutagenic of one and the other of those 2 strains, so they were necessary. In general, pre-incubations with 30% S9 produce greater mutagenic responses than with 10% S9, and in general, hamster liver S9 produce higher mutagenic responses than rat liver S9. You just heard that from the Mutamine study. The length of the pre-incubation had an effect on the small molecule nitrosamines, as was demonstrated in the literature, but it had very little effect on the NDSRIs for whatever reason. So that increasing the length of the pre-incubation had minor impact. Factors affecting the assay, and this is important, were similar for the NDSRIs and the small molecule nitrosamines, with the exception of that length of the pre-incubation factor, which I kind of gave up on some time ago. And all this is published in these 2 papers at the top of the page. The paper with my name on it only came out a few weeks ago, so you may not be aware of it. And just to point out that the guidance document at the bottom contains recommendations for how to do the enhanced Ames test, and the findings from our studies are compatible with the recommendations in this guidance document. So I'd like to move on to some studies we've done in mammalian cell, human mammalian cell assays. And I originally viewed these as being appropriate or possible follow-up assays for data generated in the Ames test, trying to avoid in vivo studies if possible somehow, but obviously we're not there yet. And we tried to concentrate our NCTR research on a couple of systems that had endogenous metabolic activity because of the known lability of the reactive derivatives of the most potent nitrosamines. We thought that might be an advantage. And actually, it turned out to be an advantage. These mammalian systems, depending on how you calculate relative potency, can be 4 orders of magnitude more sensitive to some NDSRIs than the enhanced Ames test. These are data from our laboratory. Even doing the assay with just S9 metabolic activation in TK6 cells, which is a standard in vitro mutagenesis assay, can be up to two orders of magnitude more sensitive. So there's something going on in mammalian cells that I don't really understand. It may have to do with the S9 mix we're using, which is an ancient formula I got from Abe C. and Pat O'Neill. Probably you never heard of those people, but I remember them. And anyway, I think there's some value for using these to confirm responses in the enhanced Ames test. And if I was looking at the data, I would say if you got a negative and you wanted to confirm that it was a negative from the enhanced Ames test, I mean, you could do worse than running an in vitro mutation assay on it and adding that to your weight of evidence. And in fact, if you look at the current guidance for how to test NDSRIs for safety or mutagenesis, this is somewhat expressed in that guidance. And I just put the quote from the guidance here that says, do an in vitro mammalian cell mutagenesis assay and show some metabolism data as part of your weight of evidence for adjusting a potency score that you think is too low that you got from the CPCA, and see how the regulator views that. So the two systems I'm referring to, and these are experimental systems by and large, the TK6 cell system, which is a human lymphoblastoid system. There is an assay that's assays that are OEC test guideline compliant that you can run with these cells that involve S9 activation, and we've run those, as I said just a minute ago. But we've constructed 14 derivatives of these cells, each with a different human CYP, that some of which are relevant to nitrosamine activation, and use those for doing mutagenesis experiments. And we do the standard gene tox endpoints, including HPRT and TK mutation with them, and that's, as I said, covered in test guidelines. We're also using HepRG cells, which are a human hepatic stem cell line that can be induced to differentiate into liver cells. And then, interestingly, they can be de-differentiated and stimulated to divide. So that allows you to do a lot of genetic toxicology endpoints with these cells. And these cells express endogenously a large number of human phase 1 and phase 2 enzymes. So the potential is there to get some information on risk characterization because you're looking at both activation and detoxification pathways. It's much more robust than the activation in HepG2 cells, for instance. And we found that depending on how you culture these cells, you can get even higher levels of CYP expression. And it helps to be tall. We've looked at various endpoints with these cells, including recently using error-corrected NGS to do mutagenesis, and I'm going to describe some of that. Okay, so first to start with the TK6 system, and I think in the interest of time, even though these are very pretty slides that my colleagues have put together, I'm going to skip over some of this about how the cells were generated, and it was done by lentivirus infection, but that produces a stably expressing cell line for a transgene. And we also characterize them by using mRNA expression and looking at Western blots to demonstrate that the appropriate protein was expressed in these cells. And we did some characterization of actually the enzymatic activity using appropriate substrates. Okay, so how we use these. Okay, obviously you can't, if you're going to use them to evaluate NDSRIs or any nitrosamine, you can't do mutagenesis as an endpoint. I mean, it's just not practical. So what we did was we screened NDSRIs in these cells using a relatively high throughput endpoint like flow cytometry-based micronucleus assay. And that's what's in this figure. And you can see that we get a very robust response in this one cell line that expresses 2C19. And if you can really focus your eyes on the x-axis, you can see the doses that induce this response are in the sub-micromolar range. And that's several orders of magnitude better than you see in any kind of enhanced Ames test. So that's described in this paper here. Okay, so we've gone on to screen a whole bunch, 15, I think so far, of NDSRIs using S9 activation. Let's see. It's a fancy slide, I said. It's another one from one of my colleagues. As a first cut for gene mutation, and as sort of a follow-up to the enhanced Ames test data. And you can see for this set of NDSRIs, they correspond perfectly. The big difference is that the in vitro mammalian cell assay is a lot more sensitive. I mean, it's responsive to lower doses. The low gels for these assays are a lot 10 to 100-fold lower than for the enhanced Ames test. And as I said before, I think that's pretty good indication that the negatives are really negative, at least in an in vitro assay. So let's go on over here. And then we screen these compounds, just the positive ones, it turns out, in these 15 cell lines, the 14 cell lines that we have transfected with individual CYPs. And you can see something about the CYP, the metabolic pathway that these NDSRIs are activated by. And it's often something like CYP2C19, like N-nitrosopropanolol was, but also 2B6 is also a common CYP that's utilized by a variety of NDSRIs. It could be that the selection we have here is not, is not great enough to make any firm conclusions, but at least for this collection, it seemed to be the case that these CYPs were involved, and they're not necessarily the kind of CYPs you expect for small molecule nitrosamines. So I'd like to say something about 5 minutes, something about HepRRG cells, and this work was done by Jian So, Hannah Zhu, Xiaoxing Gao, who you may recognize from Hesse, and also Jaime Miranda and Javier Ravallo, who are our house experts in error-corrected NGS. Okay, so I said HepRRG cells can be grown in essentially two different formats. One is as standard attached cell cultures submerged under media, and that's shown on the left side of this figure. And they form under proper media conditions, something that looks like primary human hepatocyte culture. But they also can be grown as spheroids, which are little aggregates of several thousand cells in these low attachment multi-well dishes shown on the right. And you can see the little dots in the wells, and those are the spheroids. And you need, Gian has very good eyesight, so she can pick these out and determine which are the good wells and the bad wells. Okay, and the significance of the spheroids is, as I said, they express higher levels of phase 1 enzymes. And here's a plot of gene expression data. The black bar is the 3D spheroids, and the open bars are the 2D cells. And you can see in every case with the phase 1 enzymes, except for one down there, 3A7, the spheroids express higher levels. And this might not look very impressive, but if you look on the y-axis, this is on a log scale, so typically it's 10-fold higher. And notice on the phase 2 side, the expression is quite similar, so that's interesting. I put little red arrows on the CYPs that might be relevant to nitrosamine activation. So we've used both 2D and 3D HepRRG cells to do genetic toxicology evaluation. This is kind of a pilot study where we set up an experiment to measure specifically gene mutation. So you can see that it involves 3 days of treatment. Under conditions, we measure toxicity using an ATP assay and the common assay to look at DNA damage. And then we put the cells into media containing human epidermal growth factor and other media adjuvants, which cause them to go into DNA synthesis. So on this, we waited several days, and we did the micronucleus assay, which I think other people in this room have probably done with this. I know other people in this room have done with these cells. And at the end of this, we also extracted genomic DNA from the cultures and used that to do error-corrected NGS. Now, the method we use is a little unusual. I would think it's not Illumina sequencing, although we've tried that also, and it works. This is with using the PacBio Hi-Fi platform, which essentially forms a rolling circle kind of synthesis situation where an enzyme will travel around and around this circle and generate a product, multiple copies of the template, so you can compare both strands of the DNA with each other multiple times and look for the real mutations rather than the artifacts that are inevitably induced by any kind of NGS type situation. And you can see with over on the right, this remember is our first experiment. With 2D cultures, we see a significant increase in mutagenesis, up to 10 millimolar per dose. Now, if you look in the bottom with 3D spheroids, you can see a much more impressive increase at the low doses. And you can see 500 micromolar produces the maximum. And one lesson from this is that you really have to watch toxicity effects in these cultures. You can really overdose the spheroids, and presumably the 2D cultures also with these chemicals, and lose your dose response very, very easily. But it's clear that the mutagenesis response was about 10-fold higher in the spheroids than the 2D cultures, and that's sort of consistent with our expectation for 2E1 expression, which is the relevant CYP for NDMA. So here's some conclusions. I'm at the end. Human CYP transfected TK6 cells provide confirmation, not transfected, transduced, to use the proper term. TK6 cells provide confirmation of enhanced Ames test findings, plus insights into the CYPs involved with metabolizing nitrosamines to mutagens. We think that some of these CYPs that we were identified and the chemicals, the nitrosamines or NDSRIs that are activated by them would make good candidates for positive controls in the enhanced Ames test. And I've tried to make that argument multiple times. I don't know how it's being accepted by others, but it makes sense to me anyway. Having intracellular metabolism, the TK6 cell system can be extraordinarily sensitive, orders of magnitude greater than the Ames test. As I said, we found it 4 orders of magnitude greater sensitivity with the endogenously expressing CYPs, but it's even 2 orders of magnitude greater using S9, and I think this might possibly be because of the S9 activation system we use, which is kind of a unique one for mammalian cells. The HepRIG system possesses both phase 1 and phase 2 metabolic enzymes at levels comparable to those in human liver in vivo. And indeed, my impression is that these cells are used for human liver metabolism studies more commonly than they are for genetic toxicology studies like we're doing it. HepRIG cells can be differentiated into a quiescent cell population or stimulated to divide so that DNA damage can be expressed as mutations or micronuclei. And HepRIG spheroid cultures are metabolically more active than attached cultures and are more sensitive to mutagenicity of nitrosamines. And I think I demonstrated that. And here are 4 papers that we recently published that describe nitrosamine results, okay? Some of those other citations during the talk were more general in nature. And I'll leave you with this aerial view of beautiful NCTR in central Arkansas, and you can see what you're missing. Okay, thank you very much.
Thank you, Dr. Heflich. Next up, we'll have Dr. Joel Bercu, who is Executive Director of Non-Clinical Safety and Pathobiology at Gilead Sciences. He will be discussing work from the HESI collaboration, specifically summarizing the in vitro Ames so-called ring trial and in vivo transgenic rodent mutation studies. Dr. Bercu.
So I want to let you know real quick, as I'm going to be presenting quite a bit of data, and some of it I had to filter out because just the length of the presentation. So anyways, I'm just going to get started. Again, thank you for the organizers for inviting me to speak today. There's been a lot of work that's been done by HESI, but I also want to highlight that this was a work that was really collaborative amongst several different institutions, including industry, academic, and regulatory agencies as well. And so I just want to let you know that there's been a lot of work that's been behind this as well. So what I'm going to talk about is I'm just going to give you a brief overview of the nitrosamine research program. We're going to talk about the ring trial, which is the enhanced Ames test as well. In vivo mutagenicity, I'm going to kind of transition from Ames to in vivo mutagenicity, application of the Ames to NDSRIs, and what to do with a positive transgenic rodent mutation assay. So this is the nitrosamine research program. Again, multiple participating programs. I listed originally all the organizations. If I listed all the names on there, I'd have to use like multiple slides. So I just want to highlight that there's many, many people that have been involved in this as well. This is kind of an overview of the nitrosamine research program and the leadership in here. And other speakers have kind of mentioned the different aspects of the HESI research program. I'm going to be focusing on the AIMS optimization and then in vivo follow-up. But I do want to highlight that there are other groups that are ongoing as well, like, for example, the in vitro approaches and QSTAR-QM, which you've kind of heard about previously as well. I do want to just highlight a little bit about the adverse outcome pathway for nitrosamines, although you've kind of heard this previously, but this is really important when we're talking about how we detect for mutagenicity as well. Again, nitrosamines in general requires metabolic activation to form into the diazonium ion, which then results in alkylation of DNA. That DNA there has to be insufficient DNA repair, which again, that could be, once you have insufficient DNA repair, you'll have mutations of critical genes, clonal expansion and cell proliferation, which leads to carcinogenicity as well. So with that said, I'm going to transition to the ring trial, because the ring trial was looking at different pathways of nitrosamine adverse outcome pathways. So the first being a mutagenic effect as well. So where you see DNA mutagenicity that is then have a signal within the AIMS assay as well. So the AIMS assay is the one thing I do want to highlight is when we started the ring trial, we didn't have a understanding on what was the current recommendations of the enhanced AIMS test. We kind of heard some presentations about it, and a lot of this was following some of the work that Bob Heflich was presented earlier as well. And so, again, the AIMS, it's actually the bacterial reverse mutation assay, but we commonly call it the AIMS. So for this purpose, I'm going to call it the AIMS because it's simpler. And here are the tester strains. A lot of these are the OECD related tester strains. There's some specifics with the E. coli PKM 101, pre-incubation for 30 minutes, rat and hamster liver-induced S9, 30% fraction of each. And then there's some guidance about solvents. Water or organic solvents, and the organic solvents have to be the lowest possible volume used. And then they mentioned 2 nitrosamine positive controls as well. With that said, I think even though we didn't have a priori knowledge about the enhanced AIMS test, I felt like our ring trial was really poised to actually answer some of the questions about how the enhanced AIMS test actually performs as well. So I do want to highlight before I go into it, what's the importance of the enhanced AIMS test in terms of NDSRA, AI outcomes. So if you look at the CPCA and the 5 categories, if you're 4 or 5, you get a limit of 1.5 microgram per day. So but if you're 1, 2, or 3, you have limits of 400, 100, or depending on the health agency, 18 to 26.5 nanograms per day. So really, if you look at from just a practical guideline or a practical aspect according to the guidelines, the enhanced AIMS test will not help you with higher values for 4 or 5, a CPCA 4 or 5. What it will help you is for higher values for 1, 2, and 3. And for a negative enhanced AIMS test, you would be able to get 1.5 microgram per day. Notice that this is very region specific. Health Canada and EMA allow for 1.5 microgram per day, whereas FDA, as Bob had mentioned, requires additional follow-up information for a weight of evidence approach. So just what compounds did we choose? So there's some differences from how we did it versus NCTR, but I think our two approaches are very complementary of each other. We selected 32 nitrosamines, 2 labs per nitrosamine, and we selected these because they had carcinogenicity data available. And so there were 11 negative compounds and 21 positive compounds for carcinogenicity. You can see here that from a CPCA category perspective, there was relatively different representation for nitrosamines. I do want to highlight as well is that there were 3 compounds that are listed as not available. In fact, we conducted this study prior to the CPCA formation. And so, but in the design of the study, we wanted to get different compound classes, even though we were testing generally small molecule nitrosamines. We did go up to probably a molecular weight about 330, but we wanted to get a little bit more diversity in terms of those. But I want to say that 3 of the compounds are considered nitroso compounds, not considered nitrosamines, but we included those because those are more drug-like impurities as well. So here are some of the strengths and limitations. Again, some of the strengths is we did have carcinogenicity data available, so that allowed you to understand performance in relation to carcinogenic outcome. We had diverse structural features tested. Many of the compounds had mutagenicity data, so we were able to kind of follow along on what the mutagenicity data was, but they weren't tested in this enhanced Ames test. And all compounds were tested in similar conditions, so we had good a priori testing methodology as well. The limitations is the carcinogenicity data is limited in terms of what you can find in the literature. So, obviously for positive compounds, it's easy to say that that's a positive compound, even if the study is not necessarily fully robust. But the negative carcinogenicity compounds, weren't the most robust studies. For example, they didn't follow current procedures for carcinogenicity, like, for example, it didn't hit the maximum tolerated dose. There were single dose studies, limited durations, and we based our conclusions based upon author calls as well. And again, I mentioned here, 3 compounds were not nitrosamines, they were nitroso compounds, but they were drug-like. So this goes into the sensitivity, specificity, and performance metrics in relation to carcinogenicity. One of the big factors that we looked at was the sensitivity of the assay. In terms of sensitivity, we looked at different parameters, such as the percent of rat versus hamster S9. So we looked at 10% rat, 30% rat, 10% hamster, and 30% hamster. The bottom line conclusion here, even though was that the Ames assay was actually was really sensitive for detection of carcinogenicity. What we did find, though, is that the hamster S9 was more sensitive than rat S9, and increasing the percentage actually increased the sensitivity. On the flip side, the specificity was generally low in the assay, so you did get more false positive responses. But in contrast, though, by increasing there wasn't much of an impact on the overall specificity with the different conditions. So even though you had increased sensitivity for hamster S9, it didn't really impact the specificity. The one thing I do want to highlight is if you just look at the confidence intervals, it's questionable whether or not, even though you did see a difference, whether or not that's statistically significant as well. And if you notice that minus S9 has a very low sensitivity, and that's to be expected since nitrosamines generally require metabolic activation. Knowing that we had the enhanced Ames test, which looked at a combination of factors, what we tried to do was we tried to look at a combination of rat and hamster. So we looked at 10% rat, 10% hamster, and minus S9, and then minus S9 and 30% rat and 30% hamster, which is probably most closely follows the enhanced Ames test performance. And what we found here was that the agreement across labs was very high. It was in the 90 percentiles. But more importantly, the sensitivity, especially if you look at the enhanced Ames test procedures, was very, very high at 93%. And so, you know, if the compound is positive, then, you know, if the compound is carcinogenic, we feel like the Ames assay would have a good chance of picking it up as well. Again, we did see lower specificity, but I think that's to be expected from what we were seeing. And actually, these results are actually fairly consistent with what we've seen from a historical database as well. The Ames assay has a very high sensitivity, but lower specificity. So more false positives, but lesser false negatives as well. So the conclusions from the ring trial, I wasn't able to show all the data here, but again, we felt like the enhanced Ames test had the highest sensitivity with minimal impact on specificity. The enhanced Ames test didn't really, again, reduce specificity compared to the standard Ames, but we did feel like its sensitivity was much higher than the traditional standard Ames. There is a correction on that last point. We did remove minus S9 compounds. So there were a series of compounds that were actually positive without metabolic activation. And what we found is by removing those compounds, we actually saw a dramatic improvement in specificity as well. I wasn't able to show that data, but we did see a dramatic impact. The other data is we found that DMSO and water were actually, there's been some criticism around DMSO. We found that it's probably more of an issue with small molecule, like the dimethyl or the diethyl, but for larger nitrosamines, we didn't really see much of an impact of DMSO. And so we felt like DMSO and water are both suitable solvents for the enhanced Ames test. Okay, so now I'm going to transition to in vivo mutagenicity. So I talked about the enhanced Ames test, and I'm going to talk a little bit about why the enhanced Ames test is so important. So what is the current impact of in vivo mutagenicity data on the current AI? And when I say in vivo mutagenicity, I'm going to specifically refer to as the transgenic rodent mutation assay. If you look at the ICHM7 guidelines, there's different categories of what you can do for a positive Ames assay to basically understand the in vivo relevance. And since these are these require metabolic activation, the transgenic rodent mutation assay in general has been the assay that I think is in general has been considered the more appropriate. Although Maik is going to be showing data later on the COMET. I'm not going to comment on the COMET assay, but he'll go into more details later. But if you look at the OECD 488 transgenic rodent mutation assay, if it's negative, there's some regional differences on how that data is treated. So if you look at Health Canada and EMA, it says that you can follow ICH Q3A, Q3B guidelines, which is considering it non-mutagenic. And then FDA says you can have greater than 1.5 microgram per day. Don't ask me how high, much higher, but it just says greater than 1.5 microgram per day. If there's a positive, currently the way I've seen things is there's really no change in the acceptable intake. One of the things that HESI has been really working hard on is how do we interpret the positive data so that we can get a better understanding of the acceptable intake, because we believe that the mutagenic potency is really important to understanding its carcinogenic potency as well. So the first thing that in terms of understanding mutagenic potency in relation to carcinogenic potency, we looked at exemplar compounds. So each of these are small molecule related nitrosamines, but they also have very comprehensive carcinogenicity data sets. if you look at NDMA and NDEA, there was 15 different dose groups tested, and most of these had, 7, 8 different dose groups. And so the dose groups that were used to derive the gold TD50s were very comprehensive. And we selected these compounds because they had various amounts of carcinogenic potencies as well. The TGR study was tested with several dose groups. So in most of the studies, there were 6 to 7 different dose groups. And the reason why we wanted to do that is, first of all, we wanted to get a no effect level, and we wanted to get a very robust dose response curve so that we could truly understand potency. And so each of these studies, I have to say, are big transgenic rodent mutation assays, but it allowed us to get a better understanding of the dose response. And these were run by different companies, and I want to thank all the companies who ran those studies. But I also want to say that each of these companies are publishing the data individually, and so each of them have agreed to. Some of the data is already out in the literature, and some of them are coming into the literature as we speak. So this is one of the bottom lines. If you look at just comparing the TD50 to the BMDL10 for transgenic rodent mutation assay, there's an extremely strong correlation. So 0.91 is the R squared. If you look at BMDL50, which is calculated, it's a potency metric from the transgenic rodent mutation assay to the TD50. And so, in my mind, using this data, you can then get to a better estimation of carcinogenic potency as well. I did want to transition. Again, I'm doing a lot of transitioning, but I wanted to, one of the questions that came up as part of the HESI work group is, okay, it's all fine and good that it has high sensitivity relative to small molecule nitrosamines, but what about NDSRIs? And obviously, we don't have carcinogenicity data for NDSRIs, but what we do have is transgenic rodent mutation assay. And so, in talking about this initiative, we had companies donate proprietary data. Now, we hid the actual compounds and structures, but they provided us information about their Ames data and then their in vivo mutagenicity data. And then we wanted to see, okay, if you had a positive or negative response in the Ames assay, how did that correlate with the transgenic rodent mutation assay? I want to highlight that we've actually gotten more data from beyond this, which I think is great. So we have even a bigger data set that we can look at as well. I also want to highlight that not all these were tested in the enhanced Ames test because, again, at the time, the enhanced Ames test data sets weren't available, our protocol wasn't available, and so it was in 10% rat and 10% hamster. We did kind of segregate this out in terms of CPCA, and you can see the molecular weights, a lot of different molecular weights. And we had a lot more negative Ames assays as well. So that's, and so that kind of gave us, more information about how did the negative Ames assay respond. We did find that in terms of our, there was one example where you had positive in the Ames, a couple examples where you had positive in the Ames assay, but negative in the transgenic rodent mutation assay. And there was one example where you had a negative in the Ames assay, enhanced Ames assay, but was positive in the TGR assay. But again, very high sensitivity, and I'm going to talk a little bit about that. Compound 22, which was negative in the enhanced Ames test, but positive in the mutagenicity assay. And I also want to highlight that this is a category 5 as well. So if you look at this, if you look at the positive TGR assays, and you compared those to small molecular weight nitrosamines, what we found was, if you look at just the potency in the TGR assay, they were significantly less potent than your small molecule nitrosamines as well. And we have more data, and it's kind of the same trend as well. I also want to highlight is that in nitrosapyrrolidine is that orange one. That's a non-cohort of concern. That would have an acceptable intake that is near equivalent to the TTC of 1.5 microgram per day. But in all the cases for the NDSRIs, they had higher BMDs than that as well. So indicating that these are likely to be of lower potency in the transgenic rodent mutation assay, and in my mind, lower potency in the carcinogenicity assay, which makes sense if you understand that these NDSRIs are more complex in structure. You see a lot more steric hindrance, and then also the fact that from a molecular standpoint, the diazonium ion compared to the overall molecule, it has a lesser contribution as well. Oh. So I did want to highlight, though, as well, if you went back to this, that this false negative compound was extremely low in terms of the carcinogenic, the transgenic rodent mutation assay. So if you compare that to NDEA or NDMA, which is at 0.03 to 0.21, its potency was at 31 milligram per kilogram per day. So even though it was a false positive, its potency is extremely low as well in the TGR assay. So again, in terms of conclusions from this is we think that the enhanced Ames test is highly sensitive for detecting the mutagenicity of nitrosamines. The EAT assay is, just checking on time, the EAT assay is highly sensitive when you correlate NDSRIs to TGR studies. And then that in vivo TGR potency is highly correlated with carcinogenic potency as well. Okay, so lastly, I'm giving you my proposal. Unfortunately, it looks like the slides kind of rolled over a little bit, but that's okay. My proposal, based on the HESI results, and I want to say this is not regulatory guideline. This is Joel's view, and it doesn't necessarily reflect the members of HESI. So I got to make all these qualifiers, right? So if you plot the line, and the reason why I highlight some of these in bold is, to me, those are kind of some interesting cutoffs. So the ones in bold are 1.5 microgram per day, okay? And I'm looking just as a simplistic view, less than, so 0.15 microgram per day, that's tenfold less than the TTC, and 15 microgram per day as greater than the TTC. There's other values here that you can look in these cutoffs as well. But if you just plotted this in terms of that correlative slope of the line, if you had a BMDL of 3, and if you remember, the N-pyrrolidine was at 2.7, which is very close to 3 milligrams per kilogram per day, that equates to an AI of 1.5 microgram per day. So if you were to use this in practice, I would say if you had a BMDL 50 greater than 3, you could have your molecule would be within the TTC range. If you're at a BMDL of, let's say, 0.5 to, and again, I have different ranges here, but if your BMDL was greater than 0.5, then your AI would be about 0.15 microgram per day. Again, looking at the slope of the line, and you can do this for different gradations depending on how you want to do this. And I think, in my mind, this is a little bit more of a conservative approach because you're looking at ranges, you're not calculating exact values, but B, it provides a little bit more of practical guidance of how you can respond to a positive in the TGR study. And if you look at this from a practical perspective as well, these are published views or published TGR studies that were done from kind of the N-nitroso oxetines and then NTTP. And you can see here, this is just data that was extracted from those papers as well. And I took out the current EMA guidelines, which for the oxetines was 100 nanograms per day. That's a read across from NNK, and NTTP, which is based upon the CPCA category 2. But then if you looked at, okay, what is the AI based upon this slope of the line and relative potency, you're looking at about 2 micrograms per day to 5 micrograms per day for NTTP. In the case of, if you look at it more on a bucketed approach, like greater than 3, et cetera, then the proposed AI would be 1.5 microgram per day. What I would say is, this I think is a very crucial part moving forward, because to me, I think the CPCA was great in terms of being able to discriminate, or sorry, as a triage approach, which of these molecules are concerned, which are not, based upon the level of exposure. But then if you exceeded the AI, then you want to do some sort of safety evaluation to then evaluate that. And so if you're negative, that's great, but then what do you do if you're positive? Current guidance is you default back to the CPCA, which I think, again, you're ignoring the biological data that you already have. And so what I'd like to see moving forward is we use that biological data to then set limits moving forward. Again, I want to highlight, these are my opinions, and doesn't necessarily reflect HESI or any regulatory guidance as well. And then that's it for my presentation. Hopefully I made it just on time, and I'll answer any questions in the panel discussion. So thanks.
Thank you very much, Dr. Bercu. Next up, we're going to have Dr. Maik Schuler, who is head of the genetic toxicology group at Pfizer, and he will talk to us about the in vivo COMET assay and how it predicts the in vivo mutagenicity of NDSRIs. Thank you.
Thank you. Thanks for the introduction. All right, so a little bit unusual to talk about COMET assay in vivo, because I don't think any of you have considered this yet. So the reason why we came up with this, or when we originally started, and we started our testing program a lot earlier than other companies, we submitted Ames data like it's outlined in ICHM7. Regulators rejected the data. We submitted read-across data. Regulator rejected our read-across substrates. So we need to come up with something new. And so what we came up with was like an in vivo testing strategy, which is much more comprehensive than just the COMET assay. And I will get to this at the end. But I will also show today is like how you could actually use the COMET assay to actually define whether or not an NDSRI is positive for mutagenicity or not. So what I'm going to do today is I'm going to give you a quick introduction to the recommended assay in ICHM7. Despite the fact that we are talking about nitrosamines, the principle of ICHM7 should still apply. I will talk a little bit about the COMET assay. We'll go and tell you what it's good at, what it's not so good at, and talk about the lesions picked up by the COMET assay, which I think is important in the context of nitrosamines. The COMET assay is not a universal tool for all genotoxic impurities, but I will demonstrate to you why I think it's really, really good for nitrosamines. And again, I will talk about DNA repair as well. I will give you two examples. One is the cohort of concern nitrosamine and nitrosomorpholine. And then I will show you an example of a mutagenic non-cohort of concern nitrosamine that has the same morpholine ring in the structure, but has vastly different potencies. And then we have tested about like 25 NDSRIs. So I will show you some general testing trends, in vivo testing trends. And I will propose a new testing strategy based on in vivo data. So if you look at like the ICHM7 guideline and the recommended assays, it's very obvious that when we talk about nitrosamines, everybody would like to do the transgenic mutation assays. Unfortunately, there's very, very limited testing availability. And so we decided actually, because of this, to actually bring the assay in-house, but that's not a possibility for most companies. So we know from our own data the PIC-A assay doesn't work because nitrosamines are really not active in the bone marrow. Neither is the blood or bone marrow micronucleus assay a good tool. The UDS assay, the unscheduled DNA synthesis assay, would be great, but nobody conducts that assay anymore. So an actually positive control for that assay is actually nitrosamine. So we looked at the COMET assay. And so again, with justification, I'm going to provide that justification, it could actually be used, but you have to justify the assay and tissue selection. So what does the COMET assay do for you? So the strength of it is really that it's applicable to really all tissues. It can be applied to different species. It can be easily integrated into toxicity studies. The advantage is it's a short duration study, which actually allows you to go to a higher MTD in the study. It has a fairly low compound requirement in comparison to a transgenic mutation assay. It's technically a lot less challenging than a TGR assay or an error-corrected sequencing assay. And again, there's a fairly large data set of compounds available, and you can actually boost the sensitivity if you desire to do so. The weakness is it's not really a mutation endpoint, it's a genotoxicity endpoint. It's repairable damage. So for example, if you remove the compound from the system, within half an hour, probably most of the damage is gone. It's not a good quantitative endpoint because again, what you're measuring is repairable damage. It can be by cellular toxicity, and some of these things have been brought out in the literature. The weakness is it's actually technically less challenging than other assays, so everybody thinks that they can actually do the assay and not having really sufficient expertise. So again, background damage varies widely, and some organs have relative high background frequencies, like for example, the GI tract and rapidly dividing tissues in general. So, with some standardization automation, it could be actually a really good assay. But again, there's a lack of standardization, and some regulatory agencies, some start with an F and end in a DA, don't like the assay at all. So, so you have to keep in mind that when you talk about the COMET assay, most people think what it picks up is single-strand break and double-strand breakage. And the way it works is that you isolate your cells, you embed them in agarose, lyse every component outside of the DNA out of it, you unwind the DNA, and then in an alkali solution, you do electrophoresis on it. And what then happens is that depending on how much DNA breakage there is, there's either very little migration, like on the top in the control, or there's a lot of damage when there's a lot of DNA breakage occurring in the cell. So little known thing about the COMET assay, it actually picks up other forms of damage, like a basic site mismatches, oxidized bases, and methylated bases. And so, and if you look at like what type of compounds induce these type of damage, you can see, so nitrosamines are alkylating agents, and you can see that a lot of the damage that's measured by the COMET assay is actually induced by alkylating agents like nitrosamines. And a lot of this stuff has to do with the repair pathway that induces intermittent DNA breakage in the DNA. So again, when you look at like the assays you can use, as more you go from the left-hand side to the right-hand side, as more predictive that would be for carcinogenicity. And so when we did our testing program, we started out with a lot of different assays. So we have the COMET assay in there, we did transgenic mutation assay, we have duplex sequencing. And that really defined a little bit more like what we were doing in the assay. So we threw kind of like a lot of assays at it. And it actually ended up being really beneficial to investigate all of these different assays. So why would the COMET assay work for nitrosamines? Actually, when you look at like some of the compounds that have been tested, every compound that was tested in the in vivo COMET assay is positive in the in vivo COMET assay. Again, it's a very small data set, it's only small molecule nitrosamines. So alkylates are generally positive in the COMET assay. And again, nitrosamines are alkylating agents. And again, for compounds like EMS, the COMET assay actually had been more sensitive than the TGR assay. And keep in mind that nitrosamines are not just inducer of gene mutations, as you saw from Bob, it actually induces micronucleus response, which is a double-strand breakage event. So it's actually a chromosomal aberration event. And so in addition to that, all the repair enzymes that I showed you are likely actually measured in the COMET assays, transient DNA breaks. And so to show you this, so this is how practically some of these damaged bases are repaired. So you get your glycosylase, cuts it out, it creates an epirinic site. You cut that out, and then you fill in the bases based on the template on the other strand, right? And so if you look at like where you have transient single strand breaks, there's a lot of like single strand breaks in there. So mechanistically, it makes a lot of sense that COMET assay should actually pick up these type of lesions. So if you look at our in vivo testing approach, what we actually do is we start with a 10-day dose ranging finding study. And what we do is, because we want to know whether or not we expect the compound to be positive or negative in the mutation assay, we incorporate the COMET assay as an initial cut on genotoxicity and hence mutagenicity. And once we have the data, we design the study in a different way. If we expect the compound to be negative in the transgenic, so it's negative in the COMET assay, we design an MTD study. So pretty much go to maximum tolerated dose and test it as high as we can in 3 doses. If the compound is positive in the COMET assay, we know the outcome of the study is going to be a positive transgenic mutation assay, and we adjust the doses. We're able, based on the COMET assay, to select doses where we go from a high mutagenic response to a non-mutagenic response. Because then what we can do is, as you see on the bottom, we can do dose response modeling and look at the mutagenic potency of these compounds. And again, this is practically how this looks like. We do actually the TK of the dose range finder on day 1 and day 10, and we do the COMET assay at the end of it. And for the Big Blue study, we do actually see 2 mutations and duplex sequencing, so a form of error-corrected sequencing. So now let me give you 2 examples on how this looks like. So you saw that show before looked at N-nitrosomorpholine. That was one of the compounds that was tested in the Ames trial. And that is a CPCA category 2 with an AI of 100 nanograms per day. The TD50, based on the Bercu paper, is 0.129 mg per kg, and it has a molecular weight of 116.12. So it's a fairly small molecule. And the first thing that happened when we put that into the Ames test, we saw this. So it was positive in pretty much like everything. And if you look at like the fold increases in TF1535, that's a 160-fold increase with 10% hamster died. So we already knew that this was going to be a really potent compound. So what we did we did the COMET assay, and we tested from 0.1 to 15 mg per kg. And what we saw is we saw a significant increase in the COMET assay starting at 1 mg per kg to 15 mg per kg. And what you're seeing in red, so these are what is called hedgehogs, and there's still some considerable controversy whether or not these are cytotoxicity-induced lesions or cells with high DNA damage. That's important because I will show you that the COMET assay is really a lousy quantitative tool. So, then we did, I'm not showing you the TGR assay data because our duplex sequencing data are clearly superior to the TGR data. And what you see there is you see these like nice dose-related increases in mutations in the liver and to some extent, to a weaker extent in the kidney. So what I want you to do is keep in mind the scale of the here. So we're going up to a mutation frequency of 200 times 10 to the minus 8. And if you look at the mutation spectrum, this is what you would expect to see with a nitrosamine. So we see a lot of like T to A and T to G mutations in the system. And with higher doses, you get more and more of these like T to G, T to A mutations. So what was a little bit surprising is, so when you look at the website, so look at the molecule, you might notice it contains exactly the same morpholine ring than N-nitrosomorpholine, right? But what you're seeing there is, so based on CPCA, it's exactly in the same category, but the molecular weight, for example, is 342.39. So it's a much higher molecular weight compound, and it looks like it's probably more sterically hindered. The first thing that we thought, like, so why did we know that this compound was different? Because when we put it in the Ames assay, this is what we saw. So remember, 160-fold, 10% S9 mix. So look at the weakness of the response, right? So we barely get over a 3-fold threshold with the response. So we already knew from that result that the compound probably is different. We put it in the Ames, in the common assay. Again, we picked up the response. Again, the dose range is a little bit different here, so it's not really a perfect comparison. So it was certainly, even this weak mutagen was picked up in the common assay really, really easily. And again, you have a lot of hedgehogs, again, induced effects in there. So when we then put it into the duplex sequencing, and I show the liver data here, is, it produced statistically significant increases in mutations. But remember, we went up to 200 at 10 mg per kg with N-nitrosomorpholine. And look, with this compound at 10 mg per kg, we see a much, much weaker response. So certainly in the in vivo testing, this is a much, much weaker mutagen. And the mutation spectrum is very, very similar. Okay. So just to reintroduce the concept again, so based on the data that we have generated in HESI, mutagenic potency equates is a good correlation between mutagenic potency and carcinogenic potency. The difference here is on the right-hand side, rather than using the gold TD50s, I put down the health authorities AIs. But you see that there's a really nice correlation with both of these type of methods. So point around it is like, if this is correct, you should be able to calculate your carcinogenic potency based on mutagenicity data. And that's what we have done here, right? So I took like all the 3 endpoints, duplex sequencing, Big Blue, and the COMET assay, and just looked at, so by dose response modeling, what is the BMDL 50? And that's the dose that produces a 50% increase over background. And what you can see is that for the mutation endpoints, duplex sequencing in Big Blue, and you can see that the duplex sequencing data are much tighter, there is a difference between the 2 compounds in mutagenic potency. And I told you that the COMET assay is not really a great quantitative tool, and that's supported here is because there's no differentiation between the 2 compounds, despite the fact that for mutagenicity, you see a totally different response. And so then if you kind of like go and put this, the confidence intervals in the system, you see that for both the TGR assay and duplex sequencing, there is quite a bit of a difference in potency between the different compounds. In the way we use it, I don't like buckets. Joe likes buckets, I don't like buckets. So the way we actually do it is, so what we do is we take the BMDL 50 for, in this case, for N-nitrosoriboxetine and divide it by the BMDL 50 for N-nitrosomorpholine. And then what we do is we multiply it with the AI for N-nitrosomorpholine that is published. And based on this, you can see that the calculated AI for N-nitrosoriboxetine would be around 25,000 nanograms per day. So that's an alternative way on how you could actually do this. So certainly for me, it's a little bit puzzling that you're regulating, because we have submitted this package to regulatory agencies. So based on the feedback we have gotten, they regulate both compounds as if they would be having the same potency. And again, related to Joel's message, we need to have a way actually to get for positive compounds to really adjust for potency. Because if not, it makes no sense to regulate both compounds in the same way. So, some other things that we actually saw. So you might be interested, so how good is the COMET assay in predicting in vivo mutagenicity? And so, again, we have tested a lot more negative compounds, we all have the positive. We only missed 1 compound. And very similar to what Joel has indicated before, that is a very, very super weak compound in a mutation assay. So again, the COMET assay is really, really good prediction for the outcome of in vivo mutagenicity. The second thing is molecular weight plays a big role. So if you look at like the compounds that we looked for in vivo mutagenicity for, if you are going beyond like 400, molecular weight of 400, it appears like that all the compounds we have tested in that molecular weight range seems to be negative. So smaller nitrosamines have a higher propensity of being in vivo mutagens than large ones. The second thing is, so we did duplex sequencing with an error-corrected sequencing technology. And what you can see here is that for the most part, both methodology correlate with each other. If there's a discrepancy, duplex sequencing seems to be the more sensitive technology. So we picked up actually quite a bit of compounds that were, so we picked up 4 compounds by duplex sequencing that we didn't see positive in the TGR assay, which can be explained by high interanimal variability. And as you can see, we missed 1 compound in the Ames test. So the Queen's ones are Ames negative compounds, and the other, and the other ones are Ames positive compounds. So how does the CPCA perform in vivo testing? So if you look at like CPCA categories 1 and 2, all of the compounds tested in those 2 categories are positive in vivo mutagenicity tests. When you go to as high you go in the CPCA category, as more negative you find in vivo, which you would probably expect, and I think we saw some earlier data where that is also fairly clear. But the crucial thing is, so how does the CPCA category perform with mutagenic potency? And the answer is not really that super well, right? So if you calculate the AI based on in vivo mutagenicity data and look at like the CPCA category is a little bit like all over the place, right? And the second thing that the CPCA, what you're seeing is like category 4 and 5, there's a lot of negative compounds in there. So negative nitrosamines really do exist. They are probably expected to be neither mutagenic nor carcinogenic. So conclusions, I think the data show that the common assay could actually be a suitable biomarker of in vivo genotoxicity for many mechanisms, with the exception that it's not really a quantitative or a mutation endpoint. So how can you all now synthesize this into like a testing strategy that is a little bit more reasonable? So again, I think at the end of the day, we might come out that an EAT-compliant Ames test with some supporting information, for example, an EAT-compliant Ames test that's negative in the CPCA category 5 is probably not a concern. If it's positive as a first step, a hepatic comet assay, again, shorter, faster, cheaper, and very, very predictive. If that's negative, you have a very unlikely scenario that this is going to be an in vivo mutagen. And I think, again, if you use ICH-M7, that would be an ICH Q3 limit. Lastly, if you do the TGR or duplex sequencing assay, if it's negative, again, you should be able to control to an ICH Q3 limit, or whatever your batch data say, depending on what regulatory agency you are in. But we should also be able to actually do dose response modeling and do a potency comparison to determine an acceptable intake for nitrosamines. And I'm going to leave it there. Thank you.
Thank you, Dr. Schuler. Next up, we will have a recorded presentation from Dr. Anna Christodoulidou. She is a senior scientific officer in the Feed Additives and Contaminants Unit of the European Food Safety Authority, also known as EFSA, and she will be addressing the role of nitrosamines in foods.
Everyone. Good evening from my side of the planet, and many thanks to the organizing committee for the invitation today. I'll be discussing nitrosamines present in food, their toxicological assessment, and the characterization of the risk they pose to human health. And nitrosamines are becoming a topic of interest in the food area, given their carcinogenicity and detection in common foods, also due to concerns over the use of the preservatives nitrates and nitrites. Before I enter this discussion, very shortly, I would like to show you the way we work in the European Food Safety Authority. EFSA is the risk assessment body of the European Union. EFSA is not in charge of risk management measures. These are defined by the European Commission. EFSA receives mandates from different sources, mainly from the European Commission, but also from the European Parliament, the Member States, applicants, or create its own mandates. The assessment is performed mainly by the scientific panels made from selected independent scientists and are prepared by scientific working groups and EFSA staff. Currently, EFSA has 11 scientific panels, the scientific committee, and 15 scientific units. The panel adopts EFSA opinions, which are published in the dedicated EFSA journal. In this particular case, EFSA has received a mandate from the European Commission to assess the risks for human health related to the presence of nitrosamines in food. The assessment of endogenous formation of nitrosamines was not included in this mandate. The reason is that during the evaluation of nitrate and nitrite by EFSA in 2017, this issue has been addressed, and therefore, in the new evaluation, EFSA focused on the exogenous production of nitrosamines. I will only give you a short summary of the conclusions of the EFSA panel on food additives on the risk to the endogenous nitrosamines. The panel evaluated the information available at the time and used a model by Health Canada in 2013 in its guidelines on drinking water in order to calculate the exposure to nitrosamines at the proposed ABI of 0.07 milligram per kilogram body weight per day of nitrite ion. This model considered also as variables the presence of nitrosable amines and the rate of in vivo nitrosation. This exposure was compared with the reference point selected as the lowest PMDL of the most potent nitrosamine detected in food, nitrosodiethylamine, and it was found that the margin of exposure is very high, indicating low concern. At this point, I would like to remind you what it is a margin of exposure. The margin of exposure is a tool used by risk assessors to assess possible safety concerns arising from the presence of substances in food and feed when it is not appropriate or possible to establish a health-based guidance value. The margin of exposure is a ratio of the dose at which a low but measurable adverse effect is observed, a reference point, such as the PMDL, which is preferably used in EFSA, and the level of exposure to the substance for a given population. The margin of exposure is not a health-based guidance value, meaning it is not a safety threshold below which risk assessors conclude that the daily intake is safe. When there is evidence of harmful effects, but not enough to confirm how much is safe, the margin of exposure tells us if current intakes are likely to be harmful or not. A low margin of exposure represents a greater risk than a higher margin of exposure. This approach is very useful in the risk assessment of substances that are genotoxic and carcinogenic. A margin of exposure of 10,000 or higher, if it is based on an animal study, will be of low concern from a public health point of view and might reasonably be considered a low priority for risk management actions. Let's go back to the exogenous formation of nitrosamines. Nitrosamines are formed primarily during the processing and preparation of food from the reaction between nitrosating agents, such as nitrite salts, and amino-based substances. In the context of food, 32 types of nitrosamines have been identified in the literature as measurable in food. They were divided based on their structure as a cyclic, or other, in order to investigate any potential structure-toxicity relationship. Detecting and quantifying nitrosamines in food is a complex process requiring sensitive analytical techniques. Current methodologies include gas and liquid chromatography combined with mass spectroscopy. Advancements in these techniques have lowered detection limits, allowing to better assess exposure levels. Nitrosamines are found in a range of products, including meats processed with nitrites and nitrates, processed fish, alcoholic and non-alcoholic beverages, but also possibly in other types of food for which data are scarce. It appears that cooking methods matter for the production of nitrosamines. Frying, grilling, and smoking have all been shown to increase nitrosamine formation, although this is based on limited information. Today's discussion is dedicated mostly to drug sources. However, nitrosamines can enter the body through smoking and diet. Smoking appears to be the major source of nitrosamines in the body, followed by dietary intake, and then comes medication. Of course, the type of nitrosamines and the mechanism of action may vary from each source. At EFSA, a combined risk assessment across these sources has not been conducted. Through the One Substance, One Assessment initiative, closer collaboration with other agencies is explored in order to advance in such areas. Let's start with the findings of this assessment regarding the toxicology of nitrosamines. Toxicokinetics and adduct formation studies in animals showed that nitrosamines are metabolized by CYP enzymes. They undergo activation and form DNA adducts, which lead to mutagenic and carcinogenic effects. The most common nitrosamines in food are mainly biotransformed by CYP2E1 and CYP2A6. Based on in vitro studies, the DNA adduct formation may occur also in human tissues, in which these CYP enzymes are present. Several studies on nitrosamines have shown positive genotoxic results in both in vitro and in vivo models. The majority of nitrosamines occurring in food, highlighted here in red, show genotoxic potential in vitro and in vivo, while no such sufficient data are available for nitrosoethylamine and nitrososarcosine. For these, the panel concluded on their genotoxic potential based on their structure, their similarity with other carcinogenic substances, and other data. Acyclic nitrosamines induce tumor formation in several mammalian species, with a majority of studies performed in rodents. Tumors were found in liver, pharynx, esophagus, for stomach, upper and lower respiratory tract, and the lung. Some cyclic nitrosamines induce tumors in the liver, respiratory, and gastrointestinal tract. Some seem not carcinogenic, while the aromatic nitrosophenylamine induces tumors in the rat urinary bladder. Epidemiological studies have linked nitrosamine exposure to cancers of the digestive tract, including gastric, colorectal, and esophageal cancers, as well as other types of cancers. The evidence points towards an association which raises further concerns about potential cancer risks in humans. However, these studies face limitations. The exposure levels are not accurately determined. There are also confounding factors, such as smoking and dietary differences, and therefore these limitations make it challenging to establish clear causal relationships. Therefore, due to the limitations in the study designs, these studies could not be used to establish tumor target sites and reference points for nitrosamines. The effects considered were acute, developmental and reproductive, genotoxic and carcinogenic. Among them, carcinogenicity is considered to be the most critical effect. From the 10 nitrosamines occurring in food and classified as carcinogenic, the lowest PMDL10 for nitrosoethylamine has been calculated as the lowest reference point. However, among these may not all share the same potency. Nitrosodimethylamine, nitrosodiethylamine, nitrosomethylethylamine, and nitrosomorpholin have been identified as having the highest carcinogenic potential. The potency of the remaining is not so clear and may affect the risk assessment approach. Therefore, two risk characterization approaches were discussed. The most conservative one, where a carcinogenic potency equal to NDEA was attributed to the other 9 nitrosamines, was the approach selected. But for comparison, a potency factor of 0.2 was considered for the 6 nitrosamines with not so clear carcinogenic potency, based on the comparison between the lowest PMDLs of the highest concern group and the lowest PMDLs of the remaining nitrosamines. EFSA receives regularly each year occurrence data from member states. In the case of nitrosamines, these data were limited for only 7 food categories and from 4 countries. Many of these were left censored, meaning below the limits of detection and quantification. In order to enrich this set of data, data were also extracted from the literature based on specified criteria. Therefore, recent EU data with reporting on left censorship and on mean concentrations per number of samples were considered. Occurrence data are not available for a number of categories. The highest values were estimated for the sum of all carcinogenic nitrosamines occurring in food for processed and unprocessed cooked meat and fish products. For the exposure estimates, 2 scenarios were considered. In scenario 1, mainly data provided to EFSA were considered, and the dietary exposure to the food carcinogenic nitrosamines was found highest for infants and toddlers with up to 55 nanogram per kilogram body weight per day. Considering that some are less carcinogenic, will reduce the value by almost 2 times. In scenario 2, all data including unprocessed cooked meat were considered, and the dietary exposure to the food carcinogenic nitrosamines was found highest for infants and toddlers with more than 200 nanogram per kilogram body weight per day. Considering that some are less carcinogenic, would reduce this value by more than 3 times. The content panel characterized the risk for scenario 1, excluding the cooked unprocessed meat and fish, and scenario 2, where the cooked unprocessed meat and fish were included. The nitrosodiethylamine PMDL10 of 10 microgram per kilogram body weight per day for increased incidence of liver tumors, benign and malignant tumors combined, in rodents was used as the reference point for the total carcinogenic nitrosamines in a margin of exposure approach. Margin of exposure values ranged at the P95 exposure in scenario 1 from more than 3,000 to 180, and in scenario 2 from 300 to 50 across dietary, surveys, and age groups. The content panel concluded that these calculated margin of exposures are below the threshold of 10,000, actually much below this threshold in both scenarios, which raises a health concern. Attributing a lower potency for some of these nitrosamines couldn't change the above conclusion. While the health risks are apparent, there are remaining uncertainties. The content panel conducted expert knowledge elicitations in order to quantify the uncertainty of the risk separately for the hazard identification and characterization and of the exposure estimates. Uncertainties in the hazard identification and characterization were not major. Uncertainties in the exposure estimates were considered important. These were the high proportion of results below the limit of detection and/or the limit of quantification, and the limited availability of data considered in the dietary exposure assessment of the sum of the carcinogenic nitrosamines. The content panel therefore noted that exposure calculations should be interpreted with caution. Despite these uncertainties, the panel concluded with very high certainty that the margin of exposure at the P95 exposure is less than 10,000 for the sum of all the nitrosamines considered in this assessment and for all age groups, which raises a health concern. In response to these findings, several recommendations have been made. The panel recommended to fill the gaps in ADME of nitrosamines relevant to human exposure, to fully characterize the metabolic activation pathways and DNA adducts formed in human and animal tissues, to perform epidemiological studies implementing a molecular approach and endorsing omics investigation on the association between nitrosamines and cancer with control of confounding factors, to standardize a sensitive analytical method to quantify the carcinogenic nitrosamines in different food products, and to collect data on nitrosamines in processed foods other than processed meat, and of products cooked in different ways with and without the addition of nitrate and nitrite. In addition, more data on human milk are needed to enable the exposure assessment in infants. In order to start addressing these recommendations and also the recommendations of the previous opinion from the panel on food additives, EFSA organized an open call for proposals related to the role of nitrate and nitrite and processing and storage in the N-nitrosamine formation in certain foods. The aim is to explore possible correlations between nitrates and nitrites and N-nitrosamine formation in food, as well as the role of cooking and storage on N-nitrosamine formation. This will be a grant of €750,000 with a deadline for submission of proposals by the end of this month and with a contract duration of 2.5 years. You can find the link in this presentation in case you are interested to apply. This is the end of my presentation, and here you can see the contributors to this opinion, the panel members, the working group members, and the EFSA staff. The opinion was published in the EFSA journal on 28 March of 2023. Thank you for your attention.
I'd like to thank our speakers for excellent and thought-provoking presentations. We've now